Right. Good afternoon, everyone. Today, in the morning session, you people have attempted grand test number 8. So once we should analyze the today's grand test question paper. Question number 91. Which of the following statements are correct about the nomenclature and taxonomy? So first one, botanical names follow ICBN rules. Botanical names follow ICBN rules, absolutely correct. ICBN stands for International Code for the Botanical Nomenclature. And number two, zoological names follow ICZN rules, International Code of Zoological Nomenclature. No ambiguity at all. Number three, taxonomy includes identification, nomenclature, and classification. Exactly. But they did not mention characterization. Taxonomy includes 4 categories, namely characterization, identification, classification, nomenclature. They didn't mention characterization, but option 3 is correct. Why? Because they did not mention only. That's why option 3 is correct. Option 4. Classification means assigning names to organisms. Wrong. Assigning names to organisms represents nomenclature, not classification. So 1, 2, 3 are correct. Answer is 1. And question number 127. The maximum number of individual of a population which can be supported with optimum resources for their survival is called carrying capacity. So it is represented as K. Either directly or indirectly, organisms perform maximum growth up to deceleration phase. Directly or indirectly, organisms perform maximum growth up to deceleration phase, called carrying capacity, beyond which further growth is not possible. Understood? So here, carrying capacity is represented as K. We can see carrying capacity in logistic growth. Logistic growth curve is demonstrated by using an equation. dN by dt is equal to Rn into K minus N by K. Understood? So answer is 2. And 128, human liver fluke, a nematode parasite, depends on which 2 intermediate hosts? Obviously snail and fish. Dear students, liver fluke is scientifically called Fasciola hepatica. So Fasciola hepatica completes its life cycle in 2 intermediate hosts. Fasciola hepatica completes its life cycle in 2 intermediate hosts, namely number 1 fish, second one is a snail. Fasciola hepatica or liver fluke completes its life cycle in 2 intermediate hosts, namely number 1 fish and second one is snail. Answer is 1, 1 and 2. Right? 129. Select the statement which explains best regarding parasitism. Obviously, in case of parasitism, organism 1 is benefited. Organism 1 is benefited, but organism 2 is affected. Benefited one is called parasite and affected one is called host. Answer is 4. Now only discussed logistic growth and exponential growth formula are respectively. Logistic growth formula dN by dt is equal to Rn into K minus N by K. And exponential growth curve model formula is dN by dt is equal to Rn. dN by dt is equal to Rn. Answer is B. 133. Western Ghats of India are considered as hotspot because they are mega biodiversity zone. Mega biodiversity zone. Answer is 2. 135. Which of the following is not a cause for biodiversity loss? Answer is 3. Keeping animals in zoological parks. If we place animals in zoological park, human beings can protect those animals. It comes under excitive conservation. But we can't see habitat loss in case of excitive conservation. So 3 is incorrect. So answer is 3. 136. Metagenesis refers to alternation between sexual and asexual generations in the life cycle. The common name of Obelia is a sea fir. The common name of Obelia is a sea fir. Obelia belongs to the phylum Nidaria or Coelenterata. Obelia performs two basic body forms, namely number 1 polyp, second one is medusa. Obelia performs two basic body forms, number 1 polyp and second one medusa. In Obelia, polyp produces medusa by asexual reproduction. Polyp produces medusa by asexual reproduction and medusa produces polyp by sexual reproduction alternately. Obelia performs two basic body forms, namely polyp and medusa. In case of Obelia, polyp produces medusa by asexual reproduction and medusa produces polyp by sexual reproduction. This phenomenon is called metagenesis or alternation of generation. So answer is 2. Diploblastic animals are exemplified by obviously Coelenterata, another name Nidaria. In Nidaria or in Coelenterata and in Ctenophora, we can see only two germ layers, namely outer ectoderm and inner endoderm. But unfortunately, in diploblastic animals, middle mesoderm is absent. Instead of mesoderm, undifferentiated layer is present called mesoglia. This is a direct question. Answer is 2. 138. Match the column. Stomachord. Stomachord is one of the characteristic feature of Belenoglasses. Belenoglasses belongs to the phylum Hemichordata. Presence of stomachord is one of the basic feature of Hemichordata. B1. Vertebral column Macropus. The common name of Macropus is kangaroo. The common name of Macropus is kangaroo. Macropus or kangaroo belongs to the class Mammalia. Macropus or kangaroo belongs to the class Mammalia. Obviously, Mammalia comes under vertebrata. Vertebrates have vertebral column. So B1, C radula. C2, radula is the basic feature of phyla. The common name of phyla is apple snail. Phyla or apple snail belongs to the phylum Mollusca. So in case of Mollusca, inside the mouth, file-like rasping organ is present, is called radula. So C2 and D3, metameric segmentation, obviously peritima. The common name of peritima is earthworm. In earthworm, body is both externally and internally divided into segments or metamers called metameric segmentation. So answer is 1. A4, B1, C2 and D3. Then 139, match the column. A, Scoliodon. Scoliodon comes under chondrichthyes, A4. And best example, trigon. The common name of trigon is stingray. So here A4Q. A, Scoliodon belongs to chondrichthyes, example Q, trigon. And B, Chelone. Chelone belongs to reptilia, B2. And example Bungarus. The common name of Bungarus is crate. So Bungarus belongs to the class reptilia. So that is why B2S. And C, Fasciola. Fasciola common name liver fluke. Just now we have discussed. Fasciola or liver fluke belongs to the phylum Platyhelminthes, C1. And example Taenia solium, tapeworm. So C1 or D, Physalia. 3, Nidaria. The common name of Physalia is Portuguese man of war. The common name of Physalia is Portuguese man of war. Physalia belongs to the phylum Nidaria or Coelenterata D3. And example P, Obelia, common name is C for just now we have discussed. So A4Q, B2S, C1R, D3P, answer is 3. Next, all up to 148 botany questions, Amma. And question number 149, vital capacity of lungs is, vital capacity of lungs is tidal volume plus inspiratory reserve volume plus expiratory reserve volume. Vital capacity is equal to tidal volume plus inspiratory reserve volume plus expiratory reserve volume. Answer is 1. And question number 150. Identify the correct set of true or false statements regarding excretory system of frog. Number 1. Frogs are uricotelic animals. Wrong. Frogs are not uricotelic. Frogs are ureotelic. So first one false. Second one, the structural and functional units of frogs' kidneys are called nephrons. Obviously correct. 2T, true. Third one, in both male and female frogs, the ureters act as common urinogenital duct. Wrong. Only in male frog, ureters act like urinogenital ducts. Why? Because in case of male frogs, ureters carry both urine and sperms. In male frogs, ureters carry both urine and sperms. Due to this reason, in male frog, we can consider ureters as urinogenital ducts. But in case of female frog, ureters carry only urine. In female frog, ureters do not carry ova. In female frog, ova will be carried by oviducts. Problem, sir? In female frog, ova will be carried by oviducts. So here, third one false. Fourth one, the urinary bladder is a thin-walled structure situated ventrally to the rectum. Obviously, urinary bladder is located ventrally to the rectum in both male and female frogs. So fourth one true, first one false, second one true, third one false, fourth one true. 151, statement 1. Cartilage possesses a hard and non-pliable matrix or ground substance rich in calcium salts that provides a structural strength. 100% wrong. Because the matrix are the ground substance of cartilage. The matrix or ground substance of cartilage contains chondroitin salts, not calcium salts. Statement 1 is incorrect. Moreover, the matrix or ground substance of cartilage is soft and pliable, not hard nature. Statement 1 is incorrect. Statement 2. Osteocytes are bone cells present in spaces called lacunae. Obviously correct. Statement 1 is incorrect, 2 is correct. 152. Tidal volume. 2. Air inspired or expired during normal breathing. A2. B. Inspiratory reserve volume. B3. Extra air or additional volume of air inspired forcibly. C1. Vital capacity. Maximum air exhaled after normal inspiration. D4. Residual volume. Air remaining in lungs after forcible expiration. So A2, B3, C1, D4. No ambiguity. Answer is 1. 153. Regarding ABO blood grouping system. Choose correct option regarding ABO blood grouping system. Number 1. Antigens are present on RBC surface. Obviously correct. Number 2. Antibodies are present in plasma. 100% correct. Fourth one. If a person with blood group B receives blood group A, it leads to agglutination. 100% correct, Amma. A person having blood group B can receive blood only from B and O. A person having blood group B can receive blood only from blood group B and O. If person with blood group B receives blood from blood group A, it leads to agglutination. 1, 2, 4 are correct. 154. During a single cardiac cycle in humans, consider the following statements regarding valve movements and pressure changes. Number 1. At the beginning of the cardiac cycle, all 4 chambers are in joint diastole and atrioventricular valves are open. Correct. Number 2. Atrial systole increases ventricular filling by about 30% without affecting semilunar valves. 100% correct. Third one, closure of atrioventricular valves during ventricular systole occurs due to rising ventricular pressure attempting backward flow of the blood into atria. No ambiguity at all, absolutely correct. Fifth one, closure of semilunar valves occur when ventricular pressure falls below atrial systole or arterial pressure, 100% correct. 1, 2, 3 and 5 are correct. 155, glycosuria, presence of glucose in urine, A4. B3, renal calculi, mass of crystallized salts within the kidney. C2, glomerulonephritis, inflammation of glomerulus. D1, gout, accumulation of uric acid crystals in joints. Answer is 3, A4, B3, C2 and D1. And 156, fill in the blanks. A, during contraction, the dash band remains constant. Obviously A band. B, the dash zone may disappear as sarcomere shortens. Obviously H zone. During muscle contraction, H zone size decreases, sometimes H zone disappears. And C, dash head binds to actin to form cross bridge. Obviously myosin head. D, ATP hydrolysis provides energy to dash the myosin heads. Activate the myosin heads. E, troponin binds to dash to expose active sites of actin. Answer, calcium ions. Answer is 1, A band, H zone, A band, B H zone, C myosin, D blank activate, E blank calcium. Answer is 1. And 157, question number 157. We have to arrange steps involved in micturition. So first one, 2. Urinary bladder fills with urine and urinary bladder becomes distended, right? Next one. Stretch receptors send signals to CNS. Next, fourth one. Same time CNS sends voluntary signals and motor messages to the wall of urinary bladder. Due to that, contractions of smooth muscles, relaxation of urethral sphincter muscles occur simultaneously. Finally, last one, urine expels out. So answer is 2, 1, 4, 3. No ambiguity, answer is 2. And next, question number 158. Arrow mark up indicates dilation or stimulation or increase. Arrow mark up indicates dilation or stimulation or increase. Arrow mark down indicates constriction or inhibition or decrease. Amma? Here P represents sympathetic neural system and Q represents parasympathetic neural system. P represents sympathetic neural system and Q represents parasympathetic neural system. In case of P, A, heart rate increases. Andre P represents sympathetic neural system. Q represents parasympathetic neural system. Here P is showing arrow mark up. Andre P, sympathetic neural system increases, heartbeat. Q, arrow mark down, means Q means parasympathetic neural system. Parasympathetic neural system decreases rate of heartbeat. Sympathetic neural system increases rate of heartbeat. Parasympathetic neural system decreases rate of heartbeat. And next one, B. B represents peristalsis, means wave-like movements in our body. P. Sympathetic neural system decreases peristalsis. Sympathetic neural system decreases peristalsis. Q. Parasympathetic neural system increases peristalsis. Actually, answer is 4. And next one, C. C means sweating. P. Sympathetic neural system increases sweating. Q. Parasympathetic neural system decreases sweating. And D represents blood pressure. Again, P. Parasympathetic neural system increases blood pressure. Q. P. Sympathetic neural system increases blood pressure. Q. Parasympathetic neural system decreases blood pressure. And E andre salivation. P. Sympathetic neural system decreases salivation. Q. Parasympathetic neural system increases salivation. F means cardiac output. P. Sympathetic neural system increases cardiac output. Q. Parasympathetic neural system decreases cardiac output. Final conclusion, listen my words again. P. Sympathetic neural system increases heart rate and parasympathetic neural system decreases heart rate. Next, peristalsis, andre wave-like movements which occur in our alimentary canal. So here, sympathetic neural system decreases peristalsis, parasympathetic neural system increases peristalsis. C. Sweating. Sympathetic neural system increases sweating, parasympathetic neural system decreases sweating. D. Blood pressure. P. Sympathetic neural system increases blood pressure. D. Parasympathetic neural system decreases blood pressure. E. Salivation. P. Sympathetic neural system decreases salivation. Q. Parasympathetic neural system increases salivation. F. Cardiac output. P. Sympathetic neural system increases cardiac output. Q. Parasympathetic neural system decreases cardiac output. Answer is 4. 159. Which of the following is true regarding corpus luteum? Answer is 3. Corpus luteum is formed after ovulation and secretes mainly progesterone. Direct answer 159, answer 3. 160. Select the incorrect statement. So incorrect option is 1. The thyroid gland plays a negligible role in the regulation of basal metabolic rate. Wrong. Thyroid gland secretes hormones like T3, triiodothyronine, T4, tetraiodothyronine or thyroxine. Both T3 and T4 play a major role in regulation of basal metabolic rate. T3 and T4 both play a major role in regulation of basal metabolic rate. Andre, the rate of oxygen consumption per unit weight of the body is called basal metabolic rate. Here they have given negligible role. Not negligible role. They play a vital role in regulation of basal metabolic rate. 1 is incorrect. 161. Adrenal glands are the glands that are present on the top of the kidneys. Each gland is located at the anterior part of the kidney. Identify the correct function of the following hormones. Mineralocorticoid function, regulate the balance of water and electrolytes. Obviously correct. Example of mineralocorticoid, aldosterone. Example of mineralocorticoid is aldosterone. Aldosterone hormone helps in reabsorption of sodium ion and water in distal convoluted tubule. Mineralocorticoid is aldosterone. Aldosterone hormone helps in joint reabsorption of sodium ion and water in distal convoluted tubule. 162, statement 1. Sertoli cells are also called nurse cells. Why do we consider the Sertoli cells as nurse cells? Because Sertoli cells provide nutrients to growing sperms. Sertoli cells are commonly called nurse cells because Sertoli cells provide nutrients to growing sperms. So here statement 1 and 2 both are correct. Answer is 1. 163. Refer the figure of mammary gland with a few structures marked as A, B, C and D. Which structure contains clusters of milk secreting cells? No ambiguity at all here. A represents clusters of cells. Clusters of cells nothing but alveoli. Clusters of cells mean alveoli. The space present in each alveolus is called lumen. Alveoli secrete milk. Milk is stored in lumen of alveoli. Answer is 1. Answer A. 164. Study the given statement and answer the question. During P phase of the menstrual cycle, during P phase of the menstrual cycle, if pregnancy does not occur, the Q withers and dies, usually around day 22 in a 28-day cycle. Obviously, luteal phase. Luteal phase is also known as secretory phase. If basically immediately after ovulation, ruptured Graafian follicle becomes corpus luteum. If fertilization does not occur, corpus luteum can survive for 14 days. After 14 days, corpus luteum degenerates and becomes corpus albicans. Q. Corpus luteum. P represents luteal phase, Q represents corpus luteum. The drop in R levels causes the lining of uterus to fall away. Obviously, R represents progesterone. This is known as, yes, menstruation. Menstruation occurs due to deficiency of progesterone. Answer is 2. P represents luteal, Q represents corpus luteum, R represents progesterone, and S represents menstruation. 165 statement 1. Successful implementation of various action plans to attain reproductive health requires strong infrastructural facilities, professional expertise, and material support. We have learnt about this in reproductive health. Statement 1 is correct. Statement 2. These are the essential to provide medical assistance and care to people in reproductive related problems. Statement 1 represents action plans. Action plans are used to treat reproductive related problems. Both the statements are true. Answer is 3. And 166, match the column. A, pill. 3, prevents ovulation. Pills prevent ovulation implantation. And pills can alter the quality of cervical mucus to prevent or to retard the entry of sperms. A3. B, condom. 1. Prevents sperms reaching the cervix. C4, vasectomy. In case of vasectomy, only seminal plasma comes out. That seminal plasma does not contain sperms. D2, IUD. Prevents implantation. Understood. Answer is 3. A3, B1, C4 and D2. 167. Choose correct or incorrect statements. A. In case of autoimmunity, the body loses its ability to distinguish self from non-self cell. Obviously correct. B. A person who has received bone marrow transplantation may develop graft versus host disease due to reaction of donor's mature T cells against host antigens. Obviously correct. For organ transplantation, tissue matching and blood group matching are essential. Otherwise, recipient's cell-mediated immune system and otherwise recipient's T lymphocytes or T cells reject donor's transplanted organs called graft rejection. D. Infants receive some degree of passive immunity through transplacental transfer of IgG antibodies from mother. Obviously, it comes under natural passive acquired immunity. A, B, D are correct, except C, all are correct. 168. The first cellular form of life appeared approximately, no ambiguity, 2000 million years ago or 2 billion years ago. Answer is 1. 169, statement 1. Adaptive radiation leads to divergent evolution. Obviously correct. Statement 2. Darwin finches are the examples of divergent evolution. No ambiguity. Both the statements are correct. Question number 170. Given below is the diagrammatic representation of living system, lymphatic system. Here A represents lymph nodes, B represents thymus, C represents lymphatic vessels. Answer is 1. A represents lymph nodes, B represents thymus, and C represents lymphatic vessels. 171. Genetic drift is more significant in small population. Genetic drift occurs in small population. Basically, in small populations, changes occur in frequency of genes or changes occur in frequency of alleles by chance but not by selection. 172. Antibodies have greatly improved our capacity to treat deadly diseases except antibiotics are available against whooping cough, diphtheria, and leprosy, but not cystic fibrosis. Answer is 4. Okay. ಈಗ remaining questions come under botany. From 173 to 180 come under botany. Thank you for your kind cooperation. All the best for tomorrow's exam. Thank you, one and all.
Ready now? So good afternoon students again. Here is the question number 8, paper number 8 in this grand test series. If you observe the question number 2, A is true and R is false. Why R is false? Enzymes are stable at high temperature and high pH, can survive in their hot springs. Their enzymes are stable at high temperature and high pH. Yes. So both are correct, but not the correct explanation. If you reverse that, assertion as reason, reason as assertion, then that will be the correct explanation. Next, 93. Choose the correct statement regarding viruses. So, the bacterial viruses, bacteriophages are using double-stranded DNA viruses. Yes, the viruses which are attacking the bacteria are going to have the double-stranded DNA, the bacteriophages, majorly. But there is one exception for this in our syllabus itself. QB bacteriophage, you can see in the molecular basis chapter, it contains the RNA as a genetic material. Next, regarding the gymnosperm life cycle, which of the following are false? So, third option is wrong. Why? Because development of pollen grains occurs outside the microsporangia. Actually, it is occur inside the microsporangium. Next, 95. Pteridophyte specimen, they are given a heart-shaped green multicellular independent structure. So, independent structure in case of the pteridophytes is both sporophyte and also gametophyte. Sporophyte is highly developed, true root, stem and leaves are there. Gametophyte is a heart-shaped structure because it is a prothallus, not having differentiation into root, stem and leaves. So, prothallus is a gametophyte of the pteridophytes. The probable option is the second one. Next, 96. Agar is used to grow microbes, correct. Agar is used in the preparation of ice creams and jellies, both are correct, but not the correct explanation. 97. Krebs cycle do not produce NAD. Krebs cycle is actually producing NADH. Next, 98. Matching, you can go through it. Next, 99. Bulliform cells which are incorrect they are asking, correct they are asking. So, they don't have any chlorophyll except that remaining 3 are correct. Chloroplast containing cells in the epidermis are going to be the, stomatal guard cells. Next, 100. So, related to the options they are given, correct option they are asking. Third and fourth are correct. In dicot leaf, parenchymatous bundle sheath cells are there. Monocot leaf cells are going to have bulliform cells. Remaining 2 are incorrect. 101. So, not correct they are asking. In dicot roots, third option is the answer. Dicot roots, hypodermis is collenchymatous they are mentioning. Which of the following is not correct? Okay. In dicot roots, hypodermis is collenchymatous. Actually, in dicot roots, the hypodermis region, entire cortex is made up of parenchyma. 102. Kelvin cycle does not require light directly because Kelvin cycle is not dependent on the light directly. It is dependent on products of light reaction, that is ATP and NADPH. The Kelvin cycle is indirectly dependent on the light because it is dependent on products of light reaction. Next, 103. So, the options they are given among the 3 are correct directly. Then a small technical issue. 105 question number. In this they are asking incorrect. So, fourth one is going to be the incorrect. Cornelius Van Neel didn't provided any evidence for the starch synthesis. He said oxygen released by green plants is from the water, not from the carbon dioxide. Next, 106. So, related to chemiosmotic, which are incorrect. Third one is incorrect. ATP is required for the proton return. Actually, in the proton return, ATP will be synthesized in the chemiosmotic hypothesis. 107. Metabolic fate of pyruvic acid during the glycolysis dependent on cellular needs and oxygen availability. So, it can undergo lactic acid fermentation, alcohol fermentation, or it is going to enter into the aerobic respiration process. 108. Related to ETS, identify the correct statement. So, first statement is the correct, you can go through it. 109. Gardener want to make plant short. So, you don't want to grow it straight higher in the height, you want to make it short. So, that he should not spray the auxin, he should spray the gibberellin, not the gibberellin, this one, cytokinin. Cytokinin is going to inhibit the apical dominance and allows the lateral shoot growth. Next, 110. Plant cell grow in size by cell enlargement and which in turn requires water, correct. Turgidity of the cells helps in the extension of the cell. Both are correct and correct explanation. 111. So that is the diagram related to Vallisneria, which is a freshwater pollinating plant. It is pollinated by water, freshwater plant, that is Vallisneria. Next, this Vallisneria female flowers will reach the surface by means of long stalk, and the male flowers were released onto the surface by detached from the mother plant body. Next, 112. So among the options they had given, double fertilization equals to syngamy plus triple fusion. Second option is correct. Next, 113. What will be the ploidy of the cells of nucellus? They had given megaspore mother cell 2n, functional megaspore nucellus 2n, functional megaspore n, embryo sac n, 2n, 2n, n and n. Next, the 114 answer is going to be 12.5. So how it is going to be? If you do it, capital T small t crossed with capital Y small, it is having like this. It is going to cross with capital T small t, capital small y. This is the logic here. In this, if you do the Punnett square by taking this, from here, as it is double heterozygous, four kinds of gametes are possible. Capital T, capital Y, capital T, small t, capital Y, small t, small y. Then here, as it is a single heterozygous, two types of gametes are formed. Capital T, small y and small t, small y. If you do this cross, you are going to get capital T, capital Y, small y, capital T, small y, capital T, small t, capital Y, small y, then capital T, small y, small y. Here, capital T, small t, capital Y, small y, capital T, small t, small y, small t. Here is going to be capital Y, small t, small y, small y. Among this, what they are asking, what proportion of phenotype could be expected to be dwarf and yellow. So, dwarf and at the same time yellow. This is the thing. So, how many are dwarf are there? This is not the thing. Here also not. This one and this one. Only two are dwarfs are there. And this is green. This is going to be yellow. Only one among this. It is going to be 12.5. If you do the percentage. Which of the following contrast states studied by Mendel? This is the third option. Next, phenotypes possible in ABO blood groups are 4. A, B, AB and O. Next, 117. Template dependent. It is going to be RNA polymerase, and primer independent they are asking. So, DNA polymerase, RNA polymerase, both are template dependent on the DNA. But DNA polymerase is primer dependent. But they are here asking primer independent, that is suitable to RNA polymerase. 118. Independent assortment occurs during meiosis. Correct. It results in the random alignment of chromosomes. Both are correct. 119. Related to linkage they are asking. Incorrect. So, third option is incorrect. Linkage increases variation. No, linkage actually decreases the variation, because linkage is inversely proportional to crossing over, and it is inversely proportional to recombination. So, because of crossing over and recombination, variations we realize. Linkage decreases the recombinations and the crossing overs, thereby the variations. Then 120. Correct related to purines. They are contain two fused heterocyclic rings, double ring structures, purines, adenine and guanine. 121. mRNA in eukaryotes generally contains both coding and non-coding sequences. Correct? In eukaryotes, genes are split into exons and introns. So, both are correct and correct explanation. Exon is going to be the coding, intron is going to be non-coding. Next, DNA fragments contain 6 repeats in an individual, 10 repeats in another. The difference is useful in DNA fingerprinting. Obviously, because number of repeats show the high degree of polymorphism, which is a basic for the DNA fingerprinting. 123, matching, direct one, you can do it off. Next, 124, charging of tRNA requires an enzyme, amino-acyl tRNA synthetase. So, it is not mentioned in our textbook, you have to note it down. Charging of tRNA is the first phase of the translation. Before the translation, it requires amino-acyl tRNA synthetase. 121, both are correct and correct explanation, genetic code, common question. 124, so A is the promoter which lies towards 5' end of the coding strand. T will be the terminator located towards 3' end. Next, 140, large central vacuole, chloroplast and cell wall. They are mentioning the characters. What is going to be? It is going to be plant cell. It is going to have abundant plasmodesmata connecting between adjacent cells. 141. So correct option they are asking. 1 and 2 are correct, ma'am. Third one is incorrect because amyloplast, elaioplast store carbohydrate, wrong. Alluroplast store oils and fats, alluro is going to be carbohydrate. Amyloplast is going to be correct, ma'am. Then 1, 2, 3. Sorry, 2 and 3 are correct, ma'am. Actually, first option will be the answer, 141. Next, 142. Meiosis 2 is an equational division, virtually identical to mitosis, correct? In both meiosis 2 and mitosis, number of chromosomes in the parent cell is reduced. So, reason is wrong. There is no reduction in the chromosome number. Assertion is going to be correct. Next, a cell 16 chromosomes in G1 phase. So, a cell with 16 chromosomes in the G1 phase they had given. Complete mitosis. How many chromosomes and DNA molecules will be there each daughter cell immediately after anaphase? So, after the anaphase they are asking. Number of chromosomes will be doubled during the anaphase. And then end of the cytokinesis. So, end of the cytokinesis, after cytokinesis, number of chromosomes will be again 16. Because it is talking about mitosis, number of chromosomes will be same. Suppose here 2C amount of DNA is there, during the S phase it will become 4C. So here also it is going to be 4C and here is going to be again 2C. Then possible option anaphase chromosome number is going to be not 22, it is 34, double 32. So anaphase 2 chromosome number 32, DNA 4C, cytokinesis 16, 2C. First option will be the answer. Next, regarding interphase, how many are correct? So except 4th one and 5th one, remaining all are correct because initial amount of DNA is donated as 2C, then it is increased to 4C. Fourth option, okay, 4th option is also correct. I think 5th option is incorrect. There is doubling in chromosome number. No, there is no doubling in chromosome number during interphase. And 6th one, initial amount of chromosome is donated at 2n and it is 4n. So both 4th, 5th and 6th are correct, incorrect. Remaining 4 are correct. 145. So it is also same related question, except 1, 2, 1, first one and the 4th one, remaining all are correct ones. Next, 146. Statement 1 is correct. Enzymes usually elevate the enzyme barrier, making the transition to S2P. Actually, enzymes decreases the activation energy so that substrate can easily reach the transition state and become the product. 2 is incorrect because they mention only one altered state. Between S and P, many altered states are there, intermediate states are there. Next, question number 148. Alkaloids is related to morphine, essential oils, lemongrass oil. You can cover it off, 148, second option. Next, directly if you go for the biotechnology part at the end, I think question number 170 plus irbodu. So 174, chromogenic substrate is used. Blue color colonies indicates non-recombinant. 174 is going to be second option they had given. Actually, blue color colonies are produced by non-recombinant. Second option. Next, why are stirred tank bioreactor prefers over shake flasks? Because it has a simple design. Next, 176. So answer will be it could multiply independently as a plasmid. If you directly inject it, it won't multiply. If you connect it with the vector and transfer into that, it will be multiplied. 177. What they are saying in the PCR. So if a researcher designs primer that annealed to the region of flask 1 kb but forgot to include DNTPs. So DNTPs are the substrates which should be added. Means the two strands of the DNA got separated. After separation, he had added the primers. Primers are going to be attached, but there is no extension going to happen. Why? Because extension is done by the deoxynucleoside triphosphates, which are not added here. So that the two strands of the DNA will be separated, but there is no extension. So 177, second option. About tissue culture, correct they are saying. So tissue culture allows large scale multiplication of plants in short time, correct. All plants produced by tissue culture are genetically identical to parent, correct, because they are clones. Tissue culture can help over virus-free plants from the infected, that is also correct, ma. Fourth one is incorrect. It is not always related to somatic hybrids. A, B, C are correct. 179. So sequence they are asking, nematode specific gene produces double stranded and mRNA silencing, nematode death, plant protection. First option correct. 180. Except third and 5 remaining all are correct. Because if you observe the third one, basmati rice varieties are only a few in number and not very diverse. Actually 20 plus varieties are there in the basmati in India. So third one is incorrect. So fifth one they had given reverse actually. Industrialized nations are rich in economy but poor in the biodiversity. So 1, 2, 4, 6 are going to be the answers for this. So there completes the discussion. Tomorrow we'll go with the paper number 9 discussion, and day after tomorrow will be the ending. Related to the admit cards, there is a issue going on. Very soon you are going to get them. Be calm, cool and hopeful for the examination. Thank you. We will meet in the tomorrow's class.
Hello all. Let's talk about grant test number 8 chemistry question paper. Here is 46th question. The first question is about tribromo octoxide. Where is tribromo octoxide is what we have, and we have to calculate the individual oxidation states. For the individual oxidation states to be calculated, we need to know the structure of tribromo octoxide, wherein the bromines which are connected to oxygens and bromine both. Here you can see one side there is bromine, but from bromine to bromine there is no electronegativity difference. As a result, there is no charge here, neither +1 nor -1. But between oxygen and bromine, oxygen is more electronegative. So the more electronegative atom always has a negative charge due to every bond that it forms with the lower electronegative atom. Andre ಈ ಹೈಯರ್ ಎಲೆಕ್ಟ್ರೋನೆಗೇಟಿವ್ ಆಟಮ್ ಲೋವರ್ ಎಲೆಕ್ಟ್ರೋನೆಗೇಟಿವ್ ಆಟಮ್ನೊಂದಿಗೆ ಫಾರ್ಮ್ ಮಾಡಿದ ಪ್ರತಿ ಬಾಂಡ್ಗೂ ಕೂಡ ಒಂದು ಪಾಸಿಟಿವ್ ಚಾರ್ಜ್ ಬರುತ್ತೆ. Therefore, ಆಕ್ಸಿಜನ್ ನೆಗೆಟಿವ್ ಚಾರ್ಜ್ ಬರುತ್ತೆ -1. ಇಲ್ಲಿ 2 ಬಾಂಡ್ಸ್ ಇರೋದ್ರಿಂದ -2, ಬ್ರೋಮಿನ್ಗೆ +2, ಮತ್ತೆ ಈ ಆಕ್ಸಿಜನ್ಗೆ -2, ಈ ಆಕ್ಸಿಜನ್ನಿಂದ ಬ್ರೋಮಿನ್ಗೆ +2, ಈ ಆಕ್ಸಿಜನ್ನಿಂದ -2, ಬ್ರೋಮಿನ್ಗೆ +2. ಅಂದರೆ ಇದರಿಂದ ಏನು ಬರಲ್ಲ, ಸೊ +6. ಇದು ಕೂಡ ಅಂತದೆ +6. ಇದರದ್ದು ನೋಡೋಣ. ಈ ಬ್ರೋಮಿನ್ನಿಂದ ಏನು ಬರಲ್ಲ, ಬಟ್ ಆಕ್ಸಿಜನ್ಸ್ರಿಂದ +2 +2 ಅಂದರೆ +4. ಸೊ 2 ಬ್ರೋಮಿನ್ಸ್ಗೆ +6, 1 ಬ್ರೋಮಿನ್ಗೆ +4 ಆಕ್ಸಿಡೇಷನ್ ಸ್ಟೇಟ್ ಬರುತ್ತೆ. ಸೊ ಆಪ್ಷನ್ ನಂಬರ್ 1 ಈಸ್ ಕರೆಕ್ಟ್ ಆನ್ಸರ್. 47. Total number of isomers possible for the compound with molecular formula C3H5BRS. ಇದು ಕನ್ಸಿಡರ್ ಮಾಡಿದ್ದಾಗ ಇದು C3H6 ಅಂದರೆ ಆಲ್ಕೀನ್ನಲ್ಲಿ ಒಂದು ಬ್ರೋಮಿನ್ ಸಬ್ಸ್ಟಿಟ್ಯೂಟ್ ಮಾಡಿದ್ದಾಗ ಹೈಡ್ರೋಜನ್ ಬದಲು ಬಂದಿರೋದು. ಆವಾಗ CH2 ಡಬಲ್ ಬಾಂಡ್ CH2 Br ಎಲಾಯ್ಲ್ ಬ್ರೋಮೈಡ್, ಅಥವಾ CH2 ಡಬಲ್ ಬಾಂಡ್ CBr CH3 ಇದು ಕೂಡ ಬರ್ಬೋದು. 2 ಬ್ರೋಮೋಪ್ರೋಪೀನ್, ಅಥವಾ ಈ ಬ್ರೋಮಿನ್ ಇಲ್ಲಿ ವಿನೈಲ್ ಬ್ರೋಮೈಡ್. ಇವು 3. ಇದು ಬಿಟ್ಟು ಸೈಕ್ಲಿಕಲ್ಲಿ ಕೂಡ C3H6 ಏನಿದೆ ಇದು ಆಲ್ಕೀನ್ಸ್ಗೆ ಸೈಕ್ಲೋ ಆಲ್ಕೇನ್ಸ್ ಕೂಡ ಐಸೋಮರ್ಸೇ. ದೇರ್ಫೋರ್ ನಾನೊಂದು ಸೈಕ್ಲೋ ಆಲ್ಕೇನ್ ಅನ್ನು ಬರೆದಿದ್ದಾರೆ, ಅದರಲ್ಲಿ ಹೈಡ್ರೋಜನ್ ತೆಗೆದು Br ಹಾಕಿದೆ, ಇಲ್ಲಿ ಕೂಡ ಹಾಕಬಹುದು ಅದನ್ನ HBr. ಈ ತರ. ದೇರ್ಫೋರ್ ಇವು ನೋಡಿದ್ದಾಗ ಸ್ಟೀರಿಯೋ ಐಸೋಮರ್ಸ್ ಅಂತಿದ್ದಾರೆ. ಸ್ಟೀರಿಯೋ ಐಸೋಮರ್ ಅಂತ ಹೇಳಿದ್ದಾಗ ಇದು ಡೆಕ್ಸ್ಟ್ರೋ ಅಂಡ್ ಲೀವೋ ಕೂಡ ನಮಗೆ ಸಾಧ್ಯತೆ ಇದೆ. ದೇರ್ಫೋರ್ ವಾಟ್ ವಿ ಕ್ಯಾನ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಹಿಯರ್ ಇಸ್ we get 1 2 3 4 5. So this is what we get. Let's go to the next part. Question number, so 5 isomers are possible. 48. When we consider here we have A, KOH, air. When we consider A, KOH and air, what we get here is which of the following is correct ಇದಕ್ಕೆ. ಸೊ ಬಿ ಅನ್ನೋ ಕಂಪೌಂಡ್ ಬಂದಿದೆ, ಸಿ ಅನ್ನುವ ಕಂಪೌಂಡ್ ಬಂದಿದೆ. ಇದು ಎಲೆಕ್ಟ್ರೋಲೈಟಿಕ್ ಆಕ್ಸಿಡೇಷನ್ ಆರ್ ಆಲ್ಕಲೈನ್ ಮೀಡಿಯಮ್ ಅಂತ ಇಫ್ ಯು ಜಸ್ಟ್ ಲುಕ್ ಎಟ್ ದಿ ಥಿಂಗ್ಸ್ ಗಿವನ್ ದೇರ್ 1 2 3 4ನಲ್ಲಿ ಒಂದು ಬ್ಲ್ಯಾಕ್ ಕೊಟ್ಟಿದ್ದಾರೆ, ಬ್ಲೂ ಕೊಟ್ಟಿದ್ದಾರೆ, ಪೊಟ್ಯಾಷಿಯಂ ಪರ್ಮ್ಯಾಂಗನೇಟ್, ಕ್ರೋಮೇಟ್ ಅದಾವೆ. ಸೊ ದ ಬ್ಯಾಕ್ ಗ್ರೌಂಡ್ ಈಸ್ ಪೊಟ್ಯಾಷಿಯಂ ಪರ್ಮ್ಯಾಂಗನೇಟ್ ಅಥವಾ ಕ್ರೋಮೇಟ್ ಅನ್ನೋದು ನಮಗೆ ಅರ್ಥ ಆಗ್ತಾ ಇದೆ. ಅದರಲ್ಲಿ ನಾವು KOH ಯೂಸ್ ಮಾಡೋದು ಇದರಲ್ಲಿ ಕೇವಲ ಪೊಟ್ಯಾಷಿಯಂ ಪರ್ಮ್ಯಾಂಗನೇಟ್ ಮ್ಯಾನುಫ್ಯಾಕ್ಚರ್ನಲ್ಲಿ. ದೇರ್ಫೋರ್ ಎ ಅನ್ನೋದು MnO2 ಆಗತ್ತೆ, ಬಿ ಅನ್ನೋದು K2MnO4 ಆಗತ್ತೆ, ಸಿ ಅನ್ನೋದು KMnO4 ಆಗತ್ತೆ. ಸೊ ಎ ಬಿಕಮ್ಸ್ MnO2, ಬಿ ಬಿಕಮ್ಸ್ K2MnO4, C becomes KMnO4. MnO2 black powder. K2MnO4 is a green compound. KMnO4 is purple. So 3, therefore option 3 is correct answer. 49. Solution of 0.1 molar ammonium hydroxide anna 0.1 molar NH4Cl ಜೊತೆ ಮಿಕ್ಸ್ ಮಾಡಿದಾಗ pH 9.25 ಬಂದಿದೆ ಅಂದರೆ the finding out then find out pKb of ammonium hydroxide ಅಂದರೆ ಅಮೋನಿಯಂ ಹೈಡ್ರಾಕ್ಸೈಡ್ ಅಮೋನಿಯಂ ಕ್ಲೋರೈಡ್ ಎರಡೂ ಕಂಬೈನ್ ಆದರೆ ಇದೊಂದು ವೀಕ್ ಬೇಸ್ ಮತ್ತು ವೀಕ್ ಬೇಸ್ ಇಸ್ ಸಾಲ್ಟ್ ವಿತ್ ಅ ಸ್ಟ್ರಾಂಗ್ ಆಸಿಡ್. ಅದಕ್ಕಾಗಿ ನಾವು ಏನು ಮಾಡ್ತಾ ಇದ್ದೀವಿ ಇದರಲ್ಲಿ ವಿ ಹ್ಯಾವ್ ಟು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ದಟ್ ಇಟ್ ಇಸ್ ಅ ಬೇಸಿಕ್ ಬಫರ್. ಬೇಸಿಕ್ ಬಫರ್ಗೆ POH equals PKB plus log of salt concentration divided by base concentration ಅಂತ ಇರುತ್ತೆ. ಇದರಲ್ಲಿ POH ಅನ್ನು ನಾವು ತಗೊಂಡಿದ್ದಾಗ pH equals 9.25 ಕೊಟ್ಟರೆ POH equals 14 minus 9.25 ಬರುತ್ತೆ, which is 4.75. 4.75 plus log salt concentration and base concentration are equal, therefore POH equals 4.75. ಅಂದರೆ ನಾನಿಲ್ಲಿ 4.75 ಇಲ್ಲಿ ಬರೀಬೇಕು. ಸೊ PKB ಅನ್ನೋದು ಇದು log 1 ಆಗುತ್ತೆ, log 1 0, POH ಅಷ್ಟೋ PKB ಅಷ್ಟೇ, ಅಂದರೆ ಇದು 4.75ಗೆ ಸಮಾನ ಆಗುತ್ತೆ. ಆಪ್ಷನ್ 2 is correct answer. 50. Chromium plus 2 is a stronger reducing agent than Fe plus 2 ion. So ಇದನ್ನು ನೋಡಿದಾಗ Cr plus 2 is a stronger reducing agent. ಯಾಕಂದರೆ Cr plus 2 ಎಲೆಕ್ಟ್ರಾನ್ ಅನ್ನು ಕಳ್ಕೊಂಡು Cr plus 3 ಆಗಿದರೆ Fe plus 2 ಎಲೆಕ್ಟ್ರಾನ್ ಅನ್ನು ಕಳ್ಕೊಂಡು Fe plus 3 ಆಗತ್ತೆ. ಇದು 3t3 ಆಗತ್ತೆ, ಇದು 3d5 ಆಗತ್ತೆ. ಇದು ಹಾಫ್ ಫಿಲ್ಡ್, ಇದು ಮೋರ್ ಸ್ಟೇಬಲ್, ಇದು 3d3 ಪಾರ್ಷಲಿ ಫಿಲ್ಡ್ ಲೆಸ್ ಸ್ಟೇಬಲ್ ಅಂತ ಅನ್ಕೋಬಾರ್ದು. ಯಾಕಂದರೆ ಇದು ಸ್ಪ್ಲಿಟ್ ಆದಾಗ ಏಕ್ವಿಯಸ್ ಸ್ಟೇಟ್ನಲ್ಲಿ ವಾಟರ್ ಆಕ್ಟ್ಸ್ ಆಸ್ ಅ ಲಿಗ್ಯಾಂಡ್. ಆವಾಗ T2G EG ಸೆಟ್ ನೋಡಿದ್ದಾಗ ಇದು T2G ಹಾಫ್ ಫಿಲ್ಡ್ ಆಗತ್ತೆ, ಇದು ಕೂಡ T2G ಹಾಫ್ ಫಿಲ್ಡ್ ಬರುತ್ತೆ, EG ಕೂಡ ಹಾಫ್ ಫಿಲ್ಡ್ ಬರುತ್ತೆ. ಆದರೂ ಕೂಡ ಇಲ್ಲಿರುವ T2G EGಗಿಂತ ಕೂಡ ಈ T2G EG ಅನ್ನೋದು ಡಿಫರೆನ್ಸ್ ಕಮ್ಮಿ ಇರುತ್ತೆ. ಅಂದರೆ ಇಲ್ಲಿ ಡೆಲ್ಟಾವೋ ಜಾಸ್ತಿ, ಇಲ್ಲಿ ಡೆಲ್ಟಾವೋ ಕಮ್ಮಿ, ಅಂದರೆ ಇಲ್ಲಿ CFSC ಜಾಸ್ತಿ, ಅಲ್ಲಿ CFSC ಕಮ್ಮಿ. ಅಂದರೆ ಇದು ಈ ರಿಯಾಕ್ಷನ್ ಮೋರ್ ಸ್ಪಾಂಟೇನಿಯಸ್ ದ್ಯಾನ್ ದಿಸ್ ಒನ್. ಇದು ಬೇಗ ಆಗತ್ತೆ, ಇದು ಸ್ಟ್ರಾಂಗರ್ ರೆಡ್ಯೂಸಿಂಗ್ ಏಜೆಂಟ್ ಆಗುತ್ತೆ ಬಿಕಾಸ್ ಆಫ್ ಹೈ ಸ್ಟೆಬಿಲಿಟಿ ಆಫ್ Cr+3 ಅಯಾನ್ ಅನ್ನೋದು ನಾವು ಅರ್ಥ ಮಾಡ್ಕೋಬೇಕು. ನೆಕ್ಸ್ಟ್ ಕ್ವೆಶ್ಚನ್, ಅಂದರೆ ಅಸರ್ಷನ್ ಕರೆಕ್ಟ್ ಇದೆ, ರೀಸನ್ ನೋಡೋಣ. Cr+3 has stable T2G half filled configuration and is E0 Cr+3 Cr+2 is negative, whereas E0 Fe+3 Fe+2 is positive. ಆ ಲಾಸ್ಟ್ ಲೈನ್ಸ್ ಕೂಡ ಕರೆಕ್ಟೇ. E0 Fe+3 Fe+2 ಅನ್ನೋವು 0.77 ಆದರೆ Cr+3 to Cr+2 ಸುಮಾರು -0.34 ಅಷ್ಟಿದೆ. ದೇರ್ಫೋರ್ ವಿ ಕ್ಯಾನ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ದಟ್ ಒನ್ ಈಸ್ ನೆಗೆಟಿವ್ ಆ್ಯಂಡ್ ಒನ್ ಈಸ್ ಪಾಸಿಟಿವ್. ಇದು ಕೂಡ ಕಾರಣನೇ. ಸೊ ಅಸರ್ಷನ್ ಇಸ್ ಟ್ರೂ, ರೀಸನ್ ಇಸ್ ಟ್ರೂ ಆ್ಯಂಡ್ ರೀಸನ್ ಎಕ್ಸ್ಪ್ಲೈನ್ಸ್ ಅಸರ್ಷನ್ ಅಂತ ತಗೋಬೇಕು. 51. The presence or absence of hydroxyl group, the presence hydroxyl group on which carbon atom or sugar differentiates RNA and DNA. ಅಂದರೆ ಕ್ವೆಶ್ಚನ್ ಏನಂದರೆ RNA DNAಗಳಲ್ಲಿ ಶುಗರ್ ಮೇಲೆ ಎಲ್ಲಿ ಹೈಡ್ರಾಕ್ಸಿ ಗ್ರೂಪ್ನಲ್ಲಿ ಆಕ್ಸಿಜನ್ ಅನ್ನೋದು ಆಬ್ಸೆಂಟ್ ಆಗಿರೋದು RNAಗೂ ಡಿಫ್ರೆನ್ಸ್ DNA-ಗೂ ಡಿಫ್ರೆನ್ಸ್ ತೋರಿಸ್ತೆ ಅಂತ. RNA ಅಂದರೆ ಏನು? ರೈಬೋಸ್ ಇರುತ್ತೆ. DNA ಫುಲ್ ಫಾರ್ಮ್ ಎಲ್ಲಿಂದ ಡಿಆಕ್ಸಿ ರೈಬೋ, ಅದು ಬೀಟಾ ಟು ಡಿಆಕ್ಸಿ, ಬೀಟಾ 2 ಡಿಆಕ್ಸಿ ಅನ್ನೋದು ಸ್ಟಾರ್ಟ್. ಅಂದರೆ ಸೆಕೆಂಡ್ ಪೊಸಿಷನ್ನಿಂದ ಡಿಆಕ್ಸಿ ತೆಗಿಬೇಕಂದ್ರೆ ಸೆಕೆಂಡ್ ಪೊಸಿಷನ್ ಅದು. ಆಪ್ಷನ್ 1 ಇಸ್ ಕರೆಕ್ಟ್ ಆನ್ಸರ್. 52. ಇನ್ Prussian blue complex, oxidation state of iron present inside and outside of the coordination sphere respectively are. Prussian blue complex ಅಂದ್ರೆ Fe4 FeCN6 times thrice ಆದರೆ ಈ ಹೊರಗಿಂದು 3 ಒಳಗಿಂದು +2 ಒಳಗಿಂದು ಫಸ್ಟ್ ಕೊಟ್ಟಿದ್ದಾರೆ ಎ ಹೊರಗಿಂದು ನೆಕ್ಸ್ಟ್ ಕೊಟ್ಟಿದ್ದಾರೆ ಕ್ವೆಶ್ಚನ್ ನಲ್ಲಿ ಬಿ therefore AB option 3 is correct answer. H2 plus Br2 gives rise to 2HBr. Then standard enthalpy of formation of HBr is minus 72.8 ಅಂತ ಅಂದರು. ಅದು ತಪ್ಪು. ಯಾಕಂದರೆ ಈಕ್ವೇಷನ್ ನಲ್ಲಿ 2 ಮೋಲ್ಸ್ ಫಾರ್ಮ್ ಆಗ್ತದವೆ. 2 ಮೋಲ್ಸ್ ಫಾರ್ಮ್ ಆಗಿದ್ರೆ ಸ್ಟ್ಯಾಂಡರ್ಡ್ ಎಂಥಾಲ್ಪಿ ಆಫ್ ಫಾರ್ಮೇಷನ್ ಅನ್ನಬಾರ್ದು. 1 ಮೋಲ್ ಫಾರ್ಮ್ ಆಗಿದ್ರೇನೆ ಹಂಗೆ ಅನ್ಬೇಕು. ಸೊ ಫಸ್ಟ್ ಸ್ಟೇಟ್ಮೆಂಟ್ ತಪ್ಪು. The coefficients in thermochemical equations may also represent number of molecules of reactants and products. ತಪ್ಪು. ಅವು ಮಾಲಿಕ್ಯೂಲ್ಸ್ ರೆಪ್ರೆಸೆಂಟ್ ಮಾಡಲ್ಲ, ಮೋಲ್ಸ್ ಮಾತ್ರ ರೆಪ್ರೆಸೆಂಟ್ ಮಾಡುತ್ತವೆ. ಯಾಕಂದರೆ ಡೆಲ್ಟಾ ಹೆಚ್ ಅನ್ನು ನಾವು ಮಾಲಿಕ್ಯೂಲ್ಸ್ ವೈಸ್ ಮೆಷರ್ ಮಾಡಲ್ಲ, ಪರ್ ಮೋಲ್ ಅಂತ ಮೆಷರ್ ಮಾಡ್ತೀವಿ. ಇದರ ಪ್ರಕಾರ ನಾನು 2 ಸ್ಟೇಟ್ಮೆಂಟ್ಸ್ ಕೂಡ ತಪ್ಪು ಅಂತ ಹೇಳ್ತಾ ಇದ್ದೀನಿ. ಆಪ್ಷನ್ 4 ಇಸ್ ಕರೆಕ್ಟ್ ಆನ್ಸರ್. 54th question. At 25 degrees Celsius and 1 atmosphere pressure, the enthalpy of combustion of benzene and acetylene are given. The change in enthalpy for reaction measure ಮಾಡಬೇಕು. ನೋಡೋಣ ಯಾವ ಥರ ಮಾಡಬೇಕು ಅನ್ನೋದು. So ಇಲ್ಲಿ at 25 degrees Celsius and 1 atmosphere pressure, the enthalpy of combustion 3C2H2 gives rise to C6H6. ಇದರಲ್ಲಿ 3C2H2 ನೋಡಿದರೆ ನಾವು ಅರ್ಥ ಮಾಡಿಕೋಬೇಕಾಗಿದೆ ಏನಂತಂದ್ರೆ ಫಸ್ಟ್ ಎಸಿಟಿಲೀನ್ ಬರ್ನ್ ಆಗಲಿ ಬೆನ್ಸಿನ್ ಬರ್ನ್ ಆಗೋದಂತ ಕೊಟ್ಟಿದ್ದಾರೆ. ಅದಕ್ಕೆ ಈಕ್ವೇಶನ್ ಬರೆಯಬೇಕು. ಅದು ಡೆಲ್ಟಾ ಹೆಚ್ ಈಕ್ವಲ್ಸ್ -3 to 68. ನೆಕ್ಸ್ಟ್ ಒನ್ ಎಸಿಟಿಲೀನ್ದು ಕೊಟ್ಟಿದ್ದಾರೆ. ದ ಚೇಂಜ್ ಇನ್ ಎಂಥಾಲ್ಪಿ ಮೆಷರ್ ಮಾಡಬೇಕು. ಇದರಿಗೋಸ್ಕರ ಹೆಸ್ ಲಾ ಅಪ್ಲೈ ಮಾಡೋಣ. ಈ ಈಕ್ವೇಷನ್ನಲ್ಲಿ C2H2 ಅನ್ನೋದು 3 ಮೋಲ್ಸ್ ಇದೆ, ಲೆಫ್ಟ್ ಸೈಡ್ ಇದೆ. ಅದೇ C2H2 ಇಲ್ಲಿದೆ, ಲೆಫ್ಟ್ ಸೈಡ್ ಇದೆ. ಸೊ 3 times 1300. ಬೆನ್ಝೀನ್ ರೈಟ್ ಸೈಡ್ ಇದೆ, 1 ಮೋಲ್ ಇದೆ. ಇದು ಲೆಫ್ಟ್ ಸೈಡ್ ಇದೆ, ರಿವರ್ಸ್ ಮಾಡಿದರೆ 3268 -3900 3268. -632 answer. So option number 4 is correct answer. 55th question. Suppose the elements X and Y combine to form compounds XY2 and X3Y2 when 0.1 mole of XY2 weighs 10 gram and 0.05 moles X3Y2 weighs 9 gram. The atomic weights of X and Y are another question. ಇದಕ್ಕೆ ಏನು ಮಾಡಬೇಕಂದ್ರೆ ನಾವು ಫಸ್ಟ್ XY2 and X3Y2 ತಗೊಳ್ಳೋಣ. XY2 ತಗೊಂಡ್ರೆ ಇದು 0.1 ಮೋಲ್ ವೇಸ್ 10 grams ಅಂದರು. ಆವಾಗ 1 ಮೋಲ್ ವೇ ಆಗೋದು ಮೋಲಾರ್ ಮಾಸ್ ಅಂದರೆ 100 grams ಆಗುತ್ತೆ. XY2 100 grams ಆಗ್ತಾ ಇದೆ ಮೋಲಾರ್ ಮಾಸ್, ವಿಚ್ ಶುಡ್ ಬಿ ದ ಸಮ್ ಆಫ್ ಅಟಾಮಿಕ್ ಮಾಸಸ್ ಆಫ್ X and Y. ಎಕ್ಸಿನ ಅಟಾಮಿಕ್ ಮಾಸ್ ಎಕ್ಸ್ ಅನ್ಕೋ, ವೈದು ಅಟಾಮಿಕ್ ಮಾಸ್ ವೈ ಅನ್ಕೋ. ಆವಾಗ ಎಕ್ಸ್ ಪ್ಲಸ್ 2y ಅಟಾಮಿಕ್ ಮಾಸ್ ಸೇರಿ 100 ಆಗಬೇಕು. ಆ ಕಡೆ 0.05 ಮೋಲ್ಸ್ ಆಫ್ X3Y2 ಇದರ ಮಾಸ್ ಬಂದ್ಬಿಟ್ಟು 9 ಗ್ರಾಮ್ ಆಗ್ತಾ ಇದೆ. ಅದರ ಟೋಟಲ್ ಮಾಸ್ ಫಸ್ಟ್ ನಾವು 1 ಮೋಲಿಗೆ ಮೆಷರ್ ಮಾಡೋಣ. 1 ಮೋಲಿಗೆ ಎಷ್ಟಾಗತ್ತೆ? ಇದು 9/20 ಅನ್ಕೊಂಡ್ರೆ ಅಂದ್ರೆ 9 × 20 ಆಗತ್ತೆ, ಅಂದ್ರೆ 180 ಆಗತ್ತೆ 1 ಮೋಲಿಗೆ. ಸೊ ಎಕ್ಸ್ದು ಮಾಸ್ ಎಕ್ಸ್, ವೈದು ಮಾಸ್ ವೈ, ಅದಕ್ಕೆ 3x + 2y = 180 ಅನ್ಕೋ. ಇದು x + 2y. ಈಗ ಎರಡು ನೋಡೋಣ. 3x + 2y is 180. x + 2y is 100. ಸಬ್ಟ್ರಾಕ್ಟ್ ಮಾಡಿದರೆ y ಇದು ಹೋಯಿತು. 2x = 80, x = 40. y ಎಷ್ಟು ಅಂದರೆ x + 40 + 2y is 100. 2y is 60, y is 30. So 40 and 30, option 1 is correct answer. Air contains 20% by volume of oxygen. The volume of air required for the complete combustion of 1 liter of methane under this condition is. ಇದಕ್ಕೆ ಮೀಥೇನ್ ತಗೊಳ್ಳಿ, ಈಕ್ವೇಶನ್ ಬರೀರಿ. ಇಷ್ಟ ಆಗತ್ತೆ. ನನಗೆ ಎಷ್ಟು ಲೀಟರ್ಸ್ ಆಫ್ ಮೀಥೇನ್ ತಗೊಂಡಾರು? 1 ಲೀಟರ್. 1 ಲೀಟರ್ ತಗೊಂಡ್ರೆ ಇದು 2 ಲೀಟರ್ ತಗೋಬೇಕು ಆಕ್ಸಿಜನ್. ಬಟ್ ಏರಿನ ವಾಲ್ಯೂಮ್ ಎಷ್ಟು ಕೊಟ್ಟಾರೆ? 20%. ಅಂದರೆ 20 ಲೀಟರ್ಸ್ ಆಕ್ಸಿಜನ್ ಆದರೆ 100 ಲೀಟರ್ಸ್ ಏರಿನ ಪರ್ಸೆಂಟೇಜ್, ಏರಿನ ವಾಲ್ಯೂಮ್. ನನಗೆ 20 ಲೀಟರ್ಸ್ ಆಫ್ ಆಕ್ಸಿಜನ್ ಬೇಕಂದ್ರೆ 100 ಲೀಟರ್ಸ್ ಆಫ್ ಏರ್ ತಗೋಬೇಕು. ಅವಾಗ 2 ಲೀಟರ್ಸ್ ತಗೋಬೇಕಂದ್ರೆ 10 ಲೀಟರ್ಸ್ ತಗೋಬೇಕು. ಸೊ ಆಪ್ಷನ್ ನಂಬರ್ 3 is correct answer. 57. A 250 milliliter aqueous solution of glucose containing 1.2 grams solute is found to be isotonic with another non-volatile solute of molecular weight 96 containing solution of 400 milliliter at 27. Then the weight of unknown solute present in second solution is. ಇದು ಐಸೋಟೋನಿಕ್ ಅಂದ್ರಲ್ವಾ, which means their osmotic pressures are same. Osmotic pressures same ಇದ್ದರೆ pi1 and pi2 are equal. So C1 and C2 will be equal. N1/V1 = N2/V2. W2/M2/V1 = W2/M2/V2. ಇಷ್ಟು ತಗೊಂಡ್ರೆ if the boiling point of a 250 ml aqueous solution of glucose containing 1.2 gram, ಅಂದರೆ ಗ್ಲೂಕೋಸಿನ ಮಾಸ್ 1.2, ಮೋಲಾರ್ ಮಾಸ್ 180, ವಾಲ್ಯೂಮ್ 250, ನಾನ್ ವಾಲಟೈಲ್ ನಾನ್ ಎಲೆಕ್ಟ್ರೋಲೈಟ್ ಸೊಲ್ಯೂಟ್ ಆಫ್ ಮಾಸ್ ಗೊತ್ತಿಲ್ಲ 96 ಗ್ರಾಮ್, ಮೋಲಾರ್ ಮಾಸ್ ಇನ್ 400 milliliter. ಇದಿಷ್ಟು. Therefore ಇದು ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡಬೇಕು. W 1.2 divided by 180 into 250 into 400 into 96 equals W. ಇಷ್ಟು ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡಬೇಕು. ಇಷ್ಟು ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡಿದರೆ $12 by 180. Now I'm just looking if we can cancel things anywhere, any way possible, but that doesn't look like that. So I'll first cancel the zeros, and then 12 into 4 is 48. 48 into 96 divided by 18 into 25. We just have to calculate it. So 96 into 48. Let me say 25 in 100. ಇದು 100 ಅಂತ ಅಪ್ರಾಕ್ಸಿಮೇಟ್ಲಿ ಅನ್ಕೊಂಡ್ರೆ 4 ಸಲ ಕ್ಯಾನ್ಸಲ್ ಆಗುತ್ತೆ. ಅಪ್ರಾಕ್ಸಿಮೇಟ್ ಕ್ಯಾಲ್ಕುಲೇಷನ್. ಸೊ 48 into 4 ಅಂದರೆ ಅಷ್ಟು ಬರುತ್ತೆ. 192 ಡಿವೈಡೆಡ್ ಬೈ 18 ಮಾಡೋಣ. ಅಪ್ರಾಕ್ಸಿಮೇಟ್ಲಿ 1 ಪಾಯಿಂಟ್ ಸಮಥಿಂಗ್ ಅಂದರೆ ಸೆಕೆಂಡ್ ಆಪ್ಷನ್ ಇಸ್ ಕರೆಕ್ಟ್. 1.06 ಬರುತ್ತೆ. ದಟ್ ಇಸ್ ಸೆಕೆಂಡ್ ಆಪ್ಷನ್ ವಿಚ್ ಇಸ್ ಕರೆಕ್ಟ್ ಆನ್ಸರ್. Next one. If the boiling point of a given aqueous solution of urea is found to be 100.416, then the freezing point of same molar aqueous solution of magnesium chloride, considering 70% dissociation, is how much? ಈ ಎರಡಕ್ಕೂ ರೇಷಿಯೋ ತಗೊಳ್ಳೋಣ, ಬೇಗ ಆಗುತ್ತೆ. ಫಸ್ಟ್ ಡೆಲ್ಟಾ ಟಿ ಬಿ ಅಂದರೆ ಕೆ ಬಿ ಇಂಟು ಎಮ್, ಅದರಲ್ಲೆಲ್ಲ ಸಬ್ಸ್ಟಿಟ್ಯೂಟ್ ಮಾಡೋಣ. ಡೆಲ್ಟಾ ಟಿ ಬಿ ಅಂದರೆ 0.416 ಬರುತ್ತೆ. ಕೆ ಬಿ ವ್ಯಾಲ್ಯೂ 0.52, ಮೊಲಾಲಿಟಿ ಎಮ್ ಅನ್ಕೋ, ಯಾಕಂದರೆ ಎಷ್ಟು ಅನ್ನೋದು ಕೊಟ್ಟಿಲ್ಲ ಅವರು. ಡೆಲ್ಟಾ ಟಿ ಎಫ್ ಯೂರಿಯಾ ಅಲ್ವಾ, I1 ಆಗತ್ತೆ. ಮತ್ತೆ ಐ ಇಂಟು ಕೆ ಎಫ್ ಇಂಟು ಎಮ್ ಬರೆದ್ರೆ ಡೆಲ್ಟಾ ಟಿ ಎಫ್ ವ್ಯಾಲ್ಯೂ ಬಂದ್ಬಿಟ್ಟು ಡಿಪ್ರೆಷನ್ ಇನ್ ಫ್ರೀಸಿಂಗ್ ಪಾಯಿಂಟ್, ಅದರ ವ್ಯಾಲ್ಯೂ ಬಂದ್ಬಿಟ್ಟು ನಮಗೆ ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡಬೇಕು ನಾವು. ಐ ಕೂಡ ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡಬೇಕು, ಯಾಕೆಂದರೆ ಆಲ್ಫಾ ವ್ಯಾಲ್ಯೂ ಕೊಟ್ಟಿದ್ದಾರೆ 70%. I-1/n-1 ಫಾರ್ ಮೆಗ್ನೀಷಿಯಂ ಕ್ಲೋರೈಡ್. ಆವಾಗ 70% ಅಲ್ವಾ, 0.7. ಮ್ಯಾಗ್ನೀಷಿಯಂ ಕ್ಲೋರೈಡ್ರಿಂದ 3 ಅಯಾನ್ಸ್ ಬರ್ತವೆ. 1.4 ಆಗ 3 - 1 is 2. I-1, therefore it goes here, that's 2.4, which is I. Therefore 2.4 and then KF is 1.86. Molality is same. Ratio ತಗೊಳ್ಳೋಣ ಎರಡಕ್ಕೂ. 0.416 divided by delta TF equals 0.52 into molality divided by 2.4 into 1.86 into molality. ಎರಡು ಮೊಲಾಲಿಟೀಸ್ ಅನ್ನ ಕ್ಯಾನ್ಸಲ್ ಮಾಡೋಣ. 0.416 into 2.4 into 1.86 divided by 0.52 equals delta TF. ಈಗ ಇದನ್ನ ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡೋದಷ್ಟೇ ಉಳಿದಿರೋದು. ಅಪ್ರಾಕ್ಸಿಮೇಟ್ಲಿ ನಾನು 2.4 ಅನ್ನು 0.52 ನಲ್ಲಿ 0.48 ಆಗತ್ತೆ ಡಬಲ್ ಮಾಡಿದರೆ. ಬಟ್ 52ಗೆ ಅಪ್ರಾಕ್ಸಿಮೇಟ್ಲಿ ಕ್ಯಾನ್ಸಲ್ ಮಾಡ್ತೀನಿ 2 times, ಅಂದರೆ ಕ್ಯಾಲ್ಕುಲೇಷನ್ ಈಸಿ ಆಗುತ್ತೆ. therefore 0.9, this is 1.86, therefore we have 9 point something, 9.3 ಬರುತ್ತೆ. And then 0.416 into 9.3. That is minus 3.571. Option number 4 is correct answer. Closest to 3.5. You can see 37688. You get 3.7 as delta TF, which means T naught F is minus 3.7. The answer which is close to minus 3.7 is fourth option. 59th question. Nickel tetracarbonyl, tetracyanide and tetrachloride are given. Tetracarbonyl nalli configuration ಬಂದ್ಬಿಟ್ಟು 3D8 configuration. 4s2, nickel 0, charge 0. 4s2 actually. Therefore ಇದು ಸ್ಟ್ರಾಂಗ್ ಫೀಲ್ಡ್ ಲಿಗ್ಯಾಂಡ್. ಸ್ಟ್ರಾಂಗ್ ಫೀಲ್ಡ್ ಲಿಗ್ಯಾಂಡ್ ಆಗಿರೋದ್ರಿಂದ E2 ಕೂಡ ಇಲ್ಲಿ ಹೋಗಿ 3D10 ಆಗುತ್ತೆ. sp3 ಖಾಲಿಯಾಗಿ sp3 ಹೈಬ್ರಿಡೈಸೇಷನ್ ಬರುತ್ತೆ, ಅಂದರೆ ಟೆಟ್ರಾಹೀಡ್ರಲ್ ಶೇಪ್ ಬರುತ್ತೆ. ನೆಕ್ಸ್ಟ್ ನಿಕೆಲ್ ಟೆಟ್ರಾಸಯನೈಡ್. ಇದರಲ್ಲಿ ನಿಕೆಲಿನ ಚಾರ್ಜ್ +2 ಇದೆ, ಅಂದರೆ 4s0 3D8 ಆಗತ್ತೆ. 4s0 3D8 ಕನ್ಸಿಡರ್ ಮಾಡಿದ್ದಾಗ ಈ 3D8 ಅನ್ನ ನಾನು ಅಬ್ಸರ್ವ್ ಮಾಡಿದರೆ ಇದು ಅರೇಂಜ್ ಮಾಡ್ಲಿಕ್ಕೆ ಬರುತ್ತೆ. therefore 1 2 3 4 ಇಷ್ಟು ಬರುತ್ತೆ. ಅವಾಗ ಇದು ಖಾಲಿ DsP2 ಹೈಬ್ರಿಡೈಸೇಷನ್, ಸ್ಕ್ವೇರ್ ಪ್ಲೇನಾರ್ ಬರುತ್ತೆ. ನಿಕೆಲ್ ಟೆಟ್ರಾ ಕ್ಲೋರೈಡ್ ತಗೊಂಡ್ರೆ ನಿಕೆಲ್ +2 ಬರುತ್ತೆ, ಮತ್ತೆ 3D8 4S0 ಬರುತ್ತೆ. ರಿಅರೇಂಜ್ ಮಾಡ್ಲಿಕ್ಕೆ ಆಗಲ್ಲ. 3D ಫುಲ್ ಇರುತ್ತೆ. sp3 ಆಗತ್ತೆ, ಟೆಟ್ರಾಹೀಡ್ರಲ್ ಆಗತ್ತೆ. ಅಂದ್ರೆ ನಿಕೆಲ್ ಟೆಟ್ರಾಕ್ಲೋರೈಡ್, ನಿಕೆಲ್ ಟೆಟ್ರಾಕಾರ್ಬೊನಿಲ್ ಎರಡೂ ಕೂಡ they are actually tetrahedral and nickel tetracyanide is square planar. So option number 3 is correct answer. Which one of the following complexes will require more equivalents of aqueous solution? ಎದರಲ್ಲಿ ಜಾಸ್ತಿ ಕ್ಲೋರೈಡ್ಸ್ ಔಟ್ಸೈಡ್ ಇದ್ರೆ ಅದು. That is option number 3. 61. The octahedral complex of a metal ion M+3 with 4 monodentate ligands L1, L2, L3 and L4. L1, L2, L3, L4 ಅನ್ನುವ 4 ಲಿಗ್ಯಾಂಡ್ಸ್ ಇದ್ರೆ ಅದರಲ್ಲಿ ಫಸ್ಟ್ ಒನ್ ಇಂದು ರೆಡ್, ಸೆಕೆಂಡ್ ಒನ್ ಇಂದು ಗ್ರೀನ್, ಮೂರನೇದು ಯೆಲ್ಲೋ, ನಾಲ್ಕನೇದು ಬ್ಲೂ. ಇವು ನಮಗೆ ವೈಲೆಟ್, ಇಂಡಿಗೋ, ಬ್ಲೂ, ಗ್ರೀನ್, ಯೆಲ್ಲೋ, ಆರೆಂಜ್, ರೆಡ್. ಟಾಪ್ ಟು ಬಾಟಮ್ ಏನಾಗತ್ತೆ? ಟಾಪ್ ಟು ಬಾಟಮ್ ಎನರ್ಜಿ ಡಿಕ್ರೀಸ್ ಆಗತ್ತೆ. ಸೊ ಇದರಲ್ಲಿ ರೆಡ್ ಅನ್ನೋದೇನಿದೆ ಅದು ಲೀಸ್ಟ್ ಎನರ್ಜಿ. ನೆಕ್ಸ್ಟ್ ಯೆಲ್ಲೋ ಬರುತ್ತೆ, ನೆಕ್ಸ್ಟ್ ಗ್ರೀನ್ ಬರುತ್ತೆ, ಹೈಯೆಸ್ಟ್ L4 ಬರುತ್ತೆ. ದೇರ್ಫೋರ್ CFSC ಪ್ರಕಾರ ಅರೇಂಜ್ ಮಾಡಿದರೆ ಆಪ್ಷನ್ 2 ಇಸ್ ಕರೆಕ್ಟ್ ಆನ್ಸರ್. ಅಲ್ಲಿ ಕೊಟ್ಟಿದ್ದ ಕಂಪೌಂಡ್ ಮೆಸಿಟೈಲ್ ಆಕ್ಸೈಡ್. ಈ ಮೆಸಿಟೈಲ್ ಆಕ್ಸೈಡಿನ ಹೆಸರು ಕೇಳ್ತಿದ್ದಾರೆ IUPAC ನೇಮ್. CH3 CH3 ಡಬಲ್ ಬಾಂಡ್ CHCO CH3. ಕೀಟೋನ್ಗೆ ಲೀಸ್ಟ್ ನಂಬರ್ ಕೊಡಬೇಕು. ಸೊ ಇಟ್ ಈಸ್ ಪೆಂಟ್. ಪೆಂಟ್ ಥರ್ಡ್ ಪೊಸಿಷನ್ ಅಲ್ಲಿ ಈನ್ 2 ಓನ್, ಫೋರ್ತ್ ಪೊಸಿಷನ್ ಅಲ್ಲಿ ಮಿಥೈಲ್. ಸೊ 4 ಮಿಥೈಲ್ ಪೆನ್ 3 and 2 on option 1. 63rd question. ಇದು ಏನ್ ಮಾಡಿದ್ದಾರೆ? ಇದಕ್ಕೆ ಐಡೋಫಾರ್ಮ್ ಹಾಕಿದ್ದಾರೆ. ಐಡೋಫಾರ್ಮ್ ಹಾಕಿದ್ದಾಗ ಐದರ್ CO CH3 ಆದರೂ ಇರಬೇಕು, ಇಲ್ಲ CHOH CH3 ಆದರೂ ಇರಬೇಕು. CO ಇದೆ, CH3 ಇಲ್ಲ, CH2 ಐತಿ. ಸೊ ಈ ಪಾರ್ಟ್ ಐಡೋಫಾರ್ಮ್ ರಿಯಾಕ್ಷನ್ ಆಗಲ್ಲ. ಬಟ್ ಇದು CHOH CH3 ಐತಿ. ಇದೇನಾಗತ್ತೆ? ಆಸ್ ಅ ಪಾರ್ಟ್ ಆಫ್ ದ ರಿಯಾಕ್ಷನ್ ಫಸ್ಟ್ ಆಕ್ಸಿಡೈಸ್ ಆಗತ್ತೆ. ಯಾವ ತರಹ? ಈ ತರ. ನೆಕ್ಸ್ಟ್ ಏನಾಗತ್ತೆ? ಈ CH3 ಹೋಗ್ಬಿಟ್ಟು ಇಟ್ ಬಿಕಮ್ಸ್ COONA, ಆಕ್ಸಿಡೈಸ್ ಆಗಿ ಐಡೋಫಾರ್ಮ್ ರಿಯಾಕ್ಷನ್ ಅಲ್ಲಿ. ನೆಕ್ಸ್ಟ್ H+ ಹಾಕಿದ್ದಾರೆ. OH ಬರುತ್ತೆ, ಕಾರ್ಬಾಕ್ಸಿಲಿಕ್ ಆಸಿಡ್. ನೆಕ್ಸ್ಟ್ ಸೋಡಾ ಲೈಮ್ ಹಾಕಿದ್ದಾರೆ. ಅವಾಗ ಇದೆಲ್ಲ ಹೋಗ್ಬಿಡುತ್ತೆ. ದೆನ್ ಬ್ಯಾರ್ ಕೀಟೋನ್ ಉಳಿದಿರುತ್ತೆ. ಆಪ್ಷನ್ 1 ಇಸ್ ಕರೆಕ್ಟ್ ಆನ್ಸರ್. ಡಿಕಾರ್ಬಾಕ್ಸಿಲೇಟಿಂಗ್ ಏಜೆಂಟ್ ಅದು. 64. ಇದು ಈ ತರಹ ಕನ್ವರ್ಟ್ ಆಗ್ಬೇಕಂದ್ರೆ C ಡಬಲ್ ಬಾಂಡ್ ಹೋಗ್ಬೇಕು, CH2 ಆಗ್ಬೇಕಂದ್ರೆ ಅದು ಎರಡೇ ರಿಯಾಕ್ಷನ್ಸ್ ರಿಂದ ಸಾಧ್ಯ ನಮಗೆ. ವಲ್ಫ್ ಕಿಶ್ನರ್ ಒಂದು, ಕ್ಲೆಮೆನ್ಸನ್ ಒಂದು. ಆದರೆ ಕ್ಲೆಮೆನ್ಸನ್ ನಲ್ಲಿ HCl ಇರುತ್ತಲ್ವಾ, ಅದಕ್ಕೆ ಕ್ಲೆಮೆನ್ಸನ್ ರಿಡಕ್ಷನ್ ನ ಆಸಿಡ್ ಸೆನ್ಸಿಟಿವ್ ಗ್ರೂಪ್ಸ್ ಇದ್ದಾಗ ಯೂಸ್ ಮಾಡಬಾರ್ದು. ಆಸಿಡ್ ಸೆನ್ಸಿಟಿವ್ ಗ್ರೂಪ್ಸ್ ಅಂದ್ರೆ ಆಲ್ಕೋಹಾಲ್ ಇರೋದು, ಆಲ್ಕೀನ್ ಇರೋದು, ಇವೆಲ್ಲ ಆಸಿಡ್ ಸೆನ್ಸಿಟಿವ್ ಗ್ರೂಪ್ಸ್ ಆಗ್ತವೆ. ಅವಿದ್ದಾಗ ಕ್ಲೆಮೆನ್ಸನ್ ಮಾಡಬಾರ್ದು. ಮತ್ತೇನ್ ಮಾಡಬೇಕು? ವಲ್ಫ್ ಕಿಶ್ನರ್ ಮಾಡಬೇಕು. ಆಪ್ಷನ್ 2. 65. Energy and radius of first bohr orbit of helium plus and lithium plus 2 are. Helium plus ಗೂ lithium plus 2 ಗೂ ಎನರ್ಜಿ ಆ್ಯಂಡ್ ರೇಡಿಯಸ್ ಫೈಂಡ್ ಔಟ್ ಮಾಡಬೇಕು. ಫಸ್ಟ್ ಎನರ್ಜೀಸ್ ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡೋಣ. ಹೀಲಿಯಂ ಪ್ಲಸ್ಗೆ 13 ಜೂಲ್ಸ್ನಲ್ಲಿ ಬೇಕಲ್ವಾ, 2.18 ಇಂಟು 10⁻⁻18/n² ಇಂಟು z² ಹಾಕಿದರೆ -2.18 ಇಂಟು 10⁻⁸ ಇಂಟು ಗ್ರೌಂಡ್ ಸ್ಟೇಟ್ದು ಬೇಕಲ್ವಾ, ಫಸ್ಟ್ ಬೋರ್ ಆರ್ಬಿಟ್, ಸೊ 1² ಇಂಟು 2² ಹಾಕಬೇಕು. -2.18 ಇಂಟು 10⁻⁻18 ಇಂಟು 4 ಬರುತ್ತೆ. ಅದು 4 2s are 8 ಅಂದರೆ ಸುಮಾರು 8.72 ಬರುತ್ತೆ. therefore 8.72 ಇಂಟು 10⁻⁻18 ಜೂಲ್ಸ್ ಫಾರ್ ಆಟಮ್ ಇದು. ಈಗ ಅದನ್ನೇ ಲಿಥಿಯಂ +2 ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡೋಣ. ಲಿಥಿಯಂ +2 ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡಿದರೆ -2.18 ಇಂಟು 10⁻⁻18 ಡಿವೈಡೆಡ್ ಬೈ 1² ಇಂಟು z² ಅಲ್ಲಿ 3² ಅಂದರೆ 9 2s are 18, ದಟ್ ಮೀನ್ಸ್ 19 ಪಾಯಿಂಟ್ ಸಮಥಿಂಗ್, -19.62 ಇಂಟು 10⁻⁻18 ಜೂಲ್ ಪರ್ ಆಟಮ್ ಬರುತ್ತೆ. ಈಗ ರೇಡಿಯಸ್ ಅನ್ನು ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡಿದರೆ Rn ಈಕ್ವಲ್ 0.529 n², 52.9 ಪಿಕೋಮೀಟರ್ಸ್ನಲ್ಲಿ ಬೇಕು. n²/z ಇದು ಅಂದರೆ 52.9 ಇಂಟು 1²/2 ಅಂದರೆ ಸುಮಾರು 26 ಅಷ್ಟು ಬರುತ್ತೆ. ಸೊ 26.45 ಪಿಕೋಮೀಟರ್. ಇಲ್ಲಿ ಅದೇ ಫಾರ್ಮುಲಾ ಹಾಕಿದರೆ 52.9 n²/z ಹಾಕಿದರೆ 52.9 ಇಂಟು n² ಇಸ್ ಫಸ್ಟ್ ಒನ್. z ಎಷ್ಟು? ಅಟಾಮಿಕ್ ನಂಬರ್ 3. So that is 17.6. So we have option number 1, which is correct answer. 66th question. The size of a microscopic particle is 1 micron and its mass is 6 into 10⁻⁻13. If its position may be measured to within 0.1% of its size, the uncertainty in velocity is. Size 1 micron, mass 6 into 10⁻⁻13. Position is measured within an accuracy of its size of 0.1% of its size. ಅಂದರೆ ಎಷ್ಟು ಬರುತ್ತೆ? ಡೆಲ್ಟಾ ಎಕ್ಸ್ ವ್ಯಾಲ್ಯೂ ಈಕ್ವಲ್ಸ್ 1 ಮೈಕ್ರಾನ್ ಅಂದರೆ 10⁻⁻6 ಮೈಕ್ರೋ ಅಂದರೆ ಇಂಟು 0.1 ಡಿವೈಡೆಡ್ ಬೈ 100 ಅಂದರೆ 10⁻⁻9. ಓಕೆ, 10⁻⁻9 ಬರುತ್ತೆ. ಇದು ಇರಲಿ, ನೆಕ್ಸ್ಟ್ ಪಾರ್ಟಿಗೆ ಹೋಗೋಣ. ವೆಲಾಸಿಟಿಗೆ ಮಾಸನ್ನು 6 ಇಂಟು 10⁻⁻13 ಇಂಟು -3 kgಗೆ ಕನ್ವರ್ಟ್ ಮಾಡೋಣ. ಇದೆಲ್ಲ ಹಾಕಿದರೆ ಫಾರ್ಮುಲಾದಲ್ಲಿ ಡೆಲ್ಟಾ ಎಕ್ಸ್ ಇಂಟು ಡೆಲ್ಟಾ ವಿ ಗ್ರೇಟರ್ ದ್ಯಾನ್ h/4πm. ಡೆಲ್ಟಾ ಎಕ್ಸ್ ಕೂಡ ಇಲ್ಲೇ ಸಬ್ಸ್ಟಿಟ್ಯೂಟ್ ಮಾಡೋಣ. 6.63 ಇಂಟು 10⁻⁻34, 4 ಇಂಟು 3.14. Mass when we consider 6 into 10⁻⁻16 into what? Into delta x 10⁻⁻9. ಇದು ಇದರಲ್ಲಿ ಸುಮಾರು 0.52 ಟೈಮ್ ಅಂದರೆ ಅವರು 6.63 ಕೊಟ್ಟಿಲ್ಲ ಹೆಚ್ ವ್ಯಾಲ್ಯೂ, 6 ಅಷ್ಟೇ ಕೊಟ್ಟಾರೆ. 6 ತಗೋ, ಕ್ಯಾಲ್ಕುಲೇಷನ್ಗೆ ಈಸಿ ಆಗುತ್ತೆ. 4 in 6 in 6 ಕ್ಯಾನ್ಸಲ್ ಆಗುತ್ತೆ. ಇದು 25 ಆಗುತ್ತೆ. 25 ಇದರಲ್ಲಿ -9 ಟೈಮ್ಸ್ ಆಗುತ್ತೆ ಕ್ಯಾನ್ಸಲೇಷನ್. 1 ಡಿವೈಡೆಡ್ ಬೈ 4 ಇಂಟು 3.14 ತಗೊಂಡ್ರೆ ಅದು ಕೂಡ ಸಬ್ಸ್ಟಿಟ್ಯೂಟ್ ಮಾಡುವ ಅವಶ್ಯಕತೆ ಇಲ್ಲ. ಇದು ಪೈ ಅಷ್ಟೇ ಹಾಕ್ಯಾರಲ್ಲಿ, 4π ಅಷ್ಟೇ. 4π ಥರಾನೇ ಇದೆ ಅಂದರೆ 10⁻⁻9/4π ಆನ್ಸರ್. ಸೊ ವಿಚ್ ಈಸ್ ಆಪ್ಷನ್ ನಂಬರ್ 1. 67th question. The acidic nature of water will be, water ಅನ್ನೋದು ಆಲ್ಕೋಹಾಲ್ಗಿಂತ ಸ್ಟ್ರಾಂಗರ್ ಆಸಿಡ್, ಬಟ್ ಮಿಥೈಲ್ ಆಲ್ಕೋಹಾಲ್ಗಿಂತ ವೀಕರ್ ಆಸಿಡ್. ಬಟ್ ಇಲ್ಲಿ ಆಲ್ಕೋಹಾಲ್ ಅಂದ್ರೆ ಓವರ್ ಆಲ್ ತಗೋಬೇಕು. ಓವರ್ ಆಲ್ ತಗೊಂಡ್ರೆ ಎಲ್ಲ ಆಲ್ಕೋಹಾಲ್ಸ್ಗಿಂತ ಸ್ಟ್ರಾಂಗರ್ ಆಸಿಡ್ ಎಕ್ಸೆಪ್ಟ್ ಮಿಥೈಲ್ ಆಲ್ಕೋಹಾಲ್, ಅದನ್ನೇ ತಗೋಬೇಕಿಲ್ಲಿ. ಸೊ ಮೋರ್ ದ್ಯಾನ್ ಆಲ್ಕೋಹಾಲ್ಸ್ ಅನ್ನೋದು ಕರೆಕ್ಟ್. ಮೋರ್ ದ್ಯಾನ್ ಫಿನಾಲ್ ತಪ್ಪು, ಫಿನಾಲ್ಗೂ ಆಲ್ಕೋಹಾಲ್ಗೂ ಮಧ್ಯದಲ್ಲಿರುತ್ತೆ. ಲೆಸ್ ದ್ಯಾನ್ ಫಿನಾಲ್ ಕರೆಕ್ಟ್, ಲೆಸ್ ದ್ಯಾನ್ ಆಲ್ಕೋಹಾಲ್ ತಪ್ಪು. Therefore A and C ಎರಡೂ ಕೂಡ ಕರೆಕ್ಟ್ ಅದಾವೆ. Select the pair in which the first ionization energy is greater than for the second ionization. ಬೆರಿಲಿಯಂ ಬೋರಾನ್ ನಲ್ಲಿ ಸೆಕೆಂಡ್ ಎಲಿಮೆಂಟ್ಗಿಂತ ಕೂಡ ಗ್ರೇಟರ್ ಫಾರ್ ದ ಸೆಕೆಂಡ್ ಎಲಿಮೆಂಟ್ಗೆ ಜಾಸ್ತಿ ಇರಬೇಕು. ಬಟ್ ಬೆರಿಲಿಯಂ ಬೋರಾನ್ ನಲ್ಲಿ ಫಸ್ಟ್ ಪಿ ನಲ್ಲಿ ಬೆರಿಲಿಯಂಗೆ ಜಾಸ್ತಿ ಇರುತ್ತೆ, ಅಂದ್ರೆ ಫಸ್ಟ್ ಎಲಿಮೆಂಟ್ಗೆ ಜಾಸ್ತಿ ಇರುತ್ತೆ, ತಗೋಬಾರ್ದು ಅದು. ಈಗ ನೆಕ್ಸ್ಟ್ ಕ್ಯೂಗೆ ಹೋದ್ರೆ ಬೋರಾನ್ ಕಾರ್ಬನ್ನಲ್ಲಿ ಕಾರ್ಬನ್ಗೆ ಜಾಸ್ತಿ ಇರುತ್ತೆ, ಸೊ ಅದು ತಗೋಬೇಕು. ಕಾರ್ಬನ್ ನೈಟ್ರೋಜನ್ಗಳಲ್ಲಿ ನೈಟ್ರೋಜನ್ಗೆ ಜಾಸ್ತಿ ಇರುತ್ತೆ, ಅದು ತಗೋಬೇಕು. ನೈಟ್ರೋಜನ್ ಆಕ್ಸಿಜನ್ಗಳಲ್ಲಿ ಆಕ್ಸಿಜನ್ಗಿಂತ ನೈಟ್ರೋಜನ್ಗೆ ಜಾಸ್ತಿ ಇರುತ್ತೆ, ಅದು ತಗೋಬಾರ್ದು. ಸೊ ಕ್ಯೂ ಅಂಡ್ ಆರ್, ಆಪ್ಷನ್ 1 ಇಸ್ ಕರೆಕ್ಟ್. ಬಾಂಡ್ ಆರ್ಡರ್ ಎಷ್ಟು ಜಾಸ್ತಿ ಇದ್ರೆ ಬಾಂಡ್ ಸ್ಟೆಬಿಲಿಟಿ ಅಷ್ಟು ಜಾಸ್ತಿ ಇರುತ್ತೆ, ಬಾಂಡ್ ಲೆಂತ್ ಅಷ್ಟು ಕಮ್ಮಿ ಇರುತ್ತೆ, ಬಾಂಡ್ ಎನರ್ಜಿ ಅಷ್ಟು ಜಾಸ್ತಿ ಇರುತ್ತೆ. ಸೊ ಫಸ್ಟ್ ಆಪ್ಷನ್ ಇಸ್ ಕರೆಕ್ಟ್. Covalent radii of atoms A and B are Ra and Rb respectively. The covalent bond length and total length of AB molecule are respectively. Covalent bond length ಅನ್ನೋದು ಈ ತರ ತಗೊಳ್ತಾರೆ. AB ಅನ್ನೋ ಆಟಮ್ಸ್ ಇದ್ರೆ ಅದು ಬಾಂಡ್ ಲೆಂತ್ ಟೋಟಲ್ ರೇಡಿಯಸ್ ತಗೊಂಡು, ಅಂದ್ರೆ ನ್ಯೂಕ್ಲಿಯಸ್ ಟು ನ್ಯೂಕ್ಲಿಯಸ್ ಡಿಸ್ಟೆನ್ಸ್ ತಗೊಂಡು ಅದರಲ್ಲಿ ಹಾಫ್ ತಗೋಬೇಕು. ಆವಾಗ ಇದರ ಕೋವಲೆಂಟ್ ರೇಡಿಯಸ್ ಆರ್ ಎ, ಅದರ ಕೋವಲೆಂಟ್ ರೇಡಿಯಸ್ ಆರ್ ಬಿ, ಅಂದ್ರೆ ಬಾಂಡ್ ಲೆಂತ್ ಬಂದ್ಬಿಟ್ಟು ಆರ್ ಎ ಪ್ಲಸ್ ಆರ್ ಬಿ. ಅದೇ ನಮಗೆ ಟೋಟಲ್ ಲೆಂತ್ ಆಫ್ ದ ಫ್ರಮ್ ಹಿಯರ್ ಟು ದೇರ್ ಬೇಕಂದ್ರೆ 2 into ಆರ್ ಎ ಪ್ಲಸ್ ಆರ್ ಬಿ. Therefore answer is option number 1. The dissolution of ionic compounds in water involves weakening of ionic interactions in presence of water. Correct? 1/80 times ಕಮ್ಮಿ ಆಗತ್ತೆ ವಾಟರ್ ನಲ್ಲಿ ಹಾಕಿದಾಗ ಎನರ್ಜಿ. ಎವಲ್ಯೂಷನ್ ಆಫ್ ಹೈಡ್ರೇಷನ್ ಎನರ್ಜಿ, ಖಂಡಿತ ಎನರ್ಜಿ ರಿಲೀಸ್ ಆಗತ್ತೆ. ಡಿಸೋಸಿಯೇಷನ್ ಕೂಡ ಆಗತ್ತೆ. ಸೊ ಆಲ್ ಆಫ್ ದೀಸ್ ಇಸ್ ಕರೆಕ್ಟ್. The following concentrations were observed at 500 Kelvin for the formation of ammonia from N2NH2 at equilibrium. We need to calculate equilibrium constant. ಸ್ವಲ್ಪ ಕ್ಯಾಲ್ಕುಲೇಷನ್ ಅನ್ನೋದು ಕಷ್ಟನೇ ಇರುತ್ತೆ ಇದಕ್ಕೆ, ಯಾಕಂದ್ರೆ Kc equals NH3 to the power 1 divided by N2 to the power half and then H2 to the power 3/2 ಅಂತ ಬರುತ್ತೆ. ಇದು Kc equals NH3 concentration at equilibrium is 1.5 into 10 to the power minus 2. N2 contribution ಅನ್ನು ತಗೊಂಡ್ರೆ N2 contribution is 2 into 10 to the power minus 2 to the power half. H2 ಇದನ್ನು ನಾವೇನ್ ಮಾಡೋಣ ಅಂದ್ರೆ we will write it as root over H2 to the power 3 ಅಂತ ಬರೆಯೋಣ. We can write it like that. ಆ ಥರ ಬರೆದ್ರೆ we have 3 into 10 to the power minus 2, ಇದಕ್ಕೆ ರೂಟ್ ಅಪ್ಲೈ ಮಾಡಿ ಹೋಲ್ ಕ್ಯೂಬ್ ಹಾಕಬೇಕು. ಇದಿಷ್ಟು ಹಾಕಿ ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡಬೇಕು. ಇದು ಕೂಡ ರೂಟೇ ಅಪ್ಲೈ ಮಾಡೋದು. 1.5 into 10 power minus 2 ರೂಟ್ 2 ಅಂದರೆ 1.414 into 10 to the power minus 2, and then we have square root of 3 andre 1.73 into 10 power minus 2 whole cube. So 1.73 2.99 ಅದು ಅಬ್ಬಾ ಸ್ಕ್ವೇರ್ ರೂಟ್ 1.73 ಅದೇ ತಾನೆ, ಅದೇ 3ನೇ ಬರುತ್ತೆ. ಸೊ 3 ಇಂಟು ತಗೊಂಡ್ರೆ ಆಯ್ತು, 3 into 1.73 ತಗೊಂಡ್ರೆ ಆಯ್ತು. $5.19 ಇದೆ ಕ್ಯೂಬ್. So Kc equals 1.5 into 10 power minus 2 by 1.41 into 10 to the power minus 2 into 5.19 into 10 to the power minus 6. Okay, square root ಇದಕ್ಕೆ square root apply ಮಾಡಿದ್ರೆ ಇದು 1 ಆಗುತ್ತೆ, ಇದು ಕೂಡ 1 ಆಗುತ್ತೆ. So 3 power 3, ಇದು ಕೂಡ 1. ಇದು 4 10 power minus 2 times. Simplify ಮಾಡಿದ್ರೆ 1.41 into 5.19. Seven point 1.5 by 7.31 into 10 power 2. If you calculate that, finally, you should get option number 2, 20 point something. 150 by 7.3 anuko. Two times already, 20 baratte approximately. So option number 2. The pH of a solution obtained by mixing those two. pH calculate ಮಾಡಬೇಕಂದ್ರೆ ಫಸ್ಟ್ A1 M1 V1 ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡ್ಕೋಬೇಕು. A1 ಅಂದರೆ ನಂಬರ್ ಆಫ್ H ಪ್ಲಸ್ ಅಯಾನ್ಸ್, ಅದು A1 M1 V1 ಈಕ್ವಲ್ಸ್ 1 ಇಂಟು ಮೊಲಾರಿಟಿ 1 ಇಂಟು ವಾಲ್ಯೂಮ್ 50 milliliter. A2 M2 V2 ತಗೋಬೇಕು, ಅದು ಕೂಡ 1 ಇಂಟು 30 ತಗೋಬೇಕು. A1 M1 V1 ಮೈನಸ್ A2 M2 V2 ಅಂದರೆ ಇದು ಜಾಸ್ತಿ ಇದೆ ಆ ಪ್ರಾಡಕ್ಟ್. ಎದರಲ್ಲಿ ಜಾಸ್ತಿ ಇರುತ್ತೆ, ಅದು ಆಯಾನ್ಸ್ ಜಾಸ್ತಿ ಇರ್ತಾವಂತ ಅರ್ಥ. ಸೊ ಡಿವೈಡೆಡ್ ಬೈ V1 ಪ್ಲಸ್ V2 ಹಾಕಿದರೆ ನಮಗೆ ಬರ್ತಿರೋದು H ಪ್ಲಸ್ ಅಯಾನ್ಸ್. ಇಲ್ಲಿ ಆಸಿಡ್ ಜಾಸ್ತಿ ಇದೆ. ಸೊ 50 ಮೈನಸ್ 30 ಡಿವೈಡೆಡ್ ಬೈ 50 ಪ್ಲಸ್ 30 ಅಂದರೆ 20 by 80, which means 1 by 4. So 0.25. 0.25 ಅಂದರೆ 2.5 into 10 power minus 1. ಈಗ pH ಅಪ್ಲೈ ಮಾಡಿದ್ರೆ ಅದಕ್ಕೆ pH equals minus log H plus minus log 2.5 into 10 power minus 1. Therefore log 2.5 plus log 10 power minus 1 minus log 2.5, which is 0.39, 0.4 anuko, minus 1, which is 0.6. Option number 1 is correct answer. 74th question. The pKa of a weak acid is 4.8. The pKb of a weak base is 4.78. The pH of an aqueous solution is. ಇದು ವೀಕ್ ಆಸಿಡ್ ವೀಕ್ ಬೇಸ್ಗಳ ಸಾಲ್ಟ್ ಅಲ್ವಾ, ಅಂದರೆ ಫಾರ್ಮುಲಾ pH ಈಕ್ವಲ್ಸ್ 7 ಪ್ಲಸ್ ಹಾಫ್ ಇಂಟು PKa ಮೈನಸ್ PKb. ಈ ಫಾರ್ಮುಲಾ ಅಪ್ಲೈ ಮಾಡಬೇಕು. 7 ಪ್ಲಸ್ ಹಾಫ್ ಇಂಟು PKa ಬಂದ್ಬಿಟ್ಟು 4.8, 4.78, 7 ಪ್ಲಸ್ ಹಾಫ್ ಇಂಟು 0.02, 7 ಪ್ಲಸ್ 7.01. That is what we get. Option 3. Next question. 75. Carbon tetrachloride can undergo hydrolysis at room temperature. Carbon tetrachloride room temperature ಅಲ್ಲಿ ಯಾವ ಟೆಂಪರೇಚರ್ ಹೈಡ್ರೋಲಿಸಿಸ್ ಆಗಲ್ಲ. ಹೈಡ್ರೋಲಿಸಿಸ್ ಆಗೋವು ಸಿಲಿಕಾನ್ ಟೆಟ್ರಾಕ್ಲೋರೈಡ್ರಿಂದ ಕೆಳಗಿನವೆಲ್ಲ. ಅಂದರೆ ಕಾರ್ಬನ್ ಟೆಟ್ರಾಕ್ಲೋರೈಡ್ ಆಗಲ್ಲ, ಸಿಲಿಕಾನ್ ಟೆಟ್ರಾಕ್ಲೋರೈಡ್, ಜರ್ಮೇನಿಯಂ ಟೆಟ್ರಾಕ್ಲೋರೈಡ್, ಟಿನ್ ಟೆಟ್ರಾಕ್ಲೋರೈಡ್, ಆಮೇಲೆ ಲೆಡ್ ಟೆಟ್ರಾಕ್ಲೋರೈಡ್, ಇವೆಲ್ಲ ಕೂಡ ಹೈಡ್ರೋಲಿಸಿಸ್ ಆಗ್ಲಿಕ್ಕೆ ಅವಕಾಶ ಇದೆ. ಬಟ್ ಕಾರ್ಬನ್ ಟೆಟ್ರಾಕ್ಲೋರೈಡ್ ಅಂತೂ ಆಗಲ್ಲ. ಯಾಕಂದರೆ ಈ ಕಾರ್ಬನ್ ಕ್ಲೋರಿನ್ ಬಾಂಡ್ ಇದರಲ್ಲಿ ಪಿ ಆರ್ಬಿಟಲ್ಸ್ ಇರ್ತವೆ. ಇದು ಹೈಡ್ರೋಲಿಸಿಸ್ ಆಗಬೇಕಂದ್ರೆ ವಾಟರ್ ಕೊಡುವ ಲೋನ್ ಪೇರನ್ನು ಆಕ್ಸೆಪ್ಟ್ ಮಾಡಬೇಕಂದ್ರೆ MTD ಆರ್ಬಿಟಲ್ಸ್ ಇರಬೇಕು. ಕಾರ್ಬನ್ನಲ್ಲಿ ಅದು ಇರಲ್ಲ ಸೆಕೆಂಡ್ ಪೀರಿಯಡ್ ಎಲಿಮೆಂಟ್ಸ್ನಲ್ಲಿ. ದೇರ್ಫೋರ್ ಅದು ತಪ್ಪು. ಡ್ಯೂರಿಂಗ್ ಹೈಡ್ರೋಲೈಸಿಸ್ ವಾಟರ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಫಾರ್ಮ್ coordinate covalent bonds with d orbitals of central atom. Correct. ಇದು ಲೋನ್ ಪೇರ್ ಕೊಡುತ್ತೆ, ಇದು ಡೋನರ್, ಅದು ಆಕ್ಸೆಪ್ಟರ್. ಸೊ there will be formation of a coordinate covalent bond. See, any tetrahalide of 14th group elements undergo hydrolysis. Not all. CCl4 does not undergo. So A and C are wrong. Therefore, option 4 is correct answer. The compound that will react with most readily with gaseous bromine andre gaseous bromine ಜೊತೆಗೆ ಬೇಗ ರಿಯಾಕ್ಟ್ ಆಗೋದು ಆಲ್ಕೀನ್ಸ್ ಫಸ್ಟ್, ಅನಂತರ ಆಲ್ಕೈನ್ಸ್, ಲಾಸ್ಟ್ಗೆ ಆಲ್ಕೇನ್ಸ್. ಸೊ C2H4 is correct answer. Acetylenic hydrogens are acidic because ಅಂದ್ರೆ ಆಲ್ಕೈನ್ ನಲ್ಲಿ ಇರುವಂತಹ ಈ ಹೈಡ್ರೋಜನ್ಸ್ ಜಾಸ್ತಿ ಅಸಿಡಿಕ್ ಎದಕ್ಕಿರ್ತವೆ ಅಂತ ಕೇಳ್ತಿದ್ದಾರೆ. ಅದು ಸಿಗ್ಮಾ ಎಲೆಕ್ಟ್ರಾನ್ ಡೆನ್ಸಿಟಿ ಆಫ್ ಕಾರ್ಬನ್ ಹೈಡ್ರೋಜನ್ ಬಾಂಡ್ in acetylene is near to carbon which has 50% S character. S character ಜಾಸ್ತಿ ಆಗಿರೋದ್ರಿಂದ ಎಲೆಕ್ಟ್ರೋನೆಗೇಟಿವಿಟಿ ಜಾಸ್ತಿ ಆಗಿ ಈ ಎಲೆಕ್ಟ್ರಾನ್ ಪೇರ್ ಇಲ್ಲಿ ಅಟ್ರಾಕ್ಟ್ ಆಗಿ ಆ ಕಾರಣದಿಂದ ಈ ತರ ಆಗ್ತಾ ಇದೆ. therefore ಅದು ಸ್ಟೇಟ್ಮೆಂಟ್ ಕರೆಕ್ಟ್ ಇದೆ. ಸಾಲ್ಯುಬಿಲಿಟಿ ಪ್ರಾಡಕ್ಟ್ ಕೊಟ್ಟಿದ್ದಾರೆ, E0 ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡಬೇಕು. ಸೊ ಇದರಿಗೋಸ್ಕರ KSP ಅನ್ನೋದು 10 to the power minus 33 ಕೊಟ್ಟರು. pH 14 ಕೊಟ್ಟಿದ್ದಾರೆ. pH 14 ಅಂದ್ರೆ POH 14 minus 14 0 ಅಂದ್ರೆ OH minus ion concentration is 1, anti-log 0 is 1. ಸೊ ಈ ತರ ನಾವು ತಗೊಂಡಿದ್ದಾಗ OH anti-log is 1, therefore OH minus ion concentration 1 molar ಆಗತ್ತೆ. ಇದರಲ್ಲಿ ನಾನು E equals E0 minus 0.059 by n log 1 by AL plus 3 concentration ಹಾಕ್ಬೇಕು ಫಾರ್ಮುಲಾ. AL plus 3 to AL ಅಂದ್ರೆ 3 electrons. ಫಸ್ಟ್ ನಾನು AL plus 3 ಕ್ಯಾಲ್ಕುಲೇಷನ್ ಮಾಡಬೇಕಂದ್ರೆ KSP equals AL plus 3 concentration into OH minus ion concentration to the power 3. Therefore, KSP is 10 power minus 33, AL plus 3 concentration, OH minus 3 ಆಗತ್ತೆ ಇದೆ. Therefore, AL plus 3 concentration is also 10 power minus 33 ಆಗತ್ತೆ. ಅಲ್ಲಿ ಸಬ್ಸ್ಟಿಟ್ಯೂಟ್ ಮಾಡಿದರೆ E equals E0 ವ್ಯಾಲ್ಯೂ ಅವರೇ ಕೊಟ್ಟಿದ್ದಾರೆ, minus 1.66. ಇದು 0.6 ಅನ್ಕೊಂಡ್ರೆ ಇದು 3 ಅನ್ಕೊಂಡ್ರೆ log 1 by minus 10 power 33 ಹಾಕಿದರೆ minus 1.66 minus 0.06 by 3, then this goes to the numerator and 33 remains, so that is 11. That is minus 1.66 minus 0.66. If you combine both of them, you'll get option number 2 is correct answer, minus 2.31 volt. 79th question. How long a current of 1.56 ampere has to be passed through aqueous solution of silver nitrate to coat a metal surface of 80 centimeter square? ಸೊ ಇದರಿಗೋಸ್ಕರ ನಾನೇನು ಮಾತಾಡ್ತಾ ಇದ್ದೀನಿ ಅಂದರೆ we are considering silver nitrate to coat a metal surface of 80 centimeter square ಅಂತ ಇದ್ವಿ. ಅದರಿಗೋಸ್ಕರ ಟೈಮ್ ಬೇಕಲ್ವಾ. ಸೊ ಫಸ್ಟ್ ನಾವು ಎಷ್ಟು ವೆಯ್ಟ್ ಅನ್ನೋದು ಮೆಷರ್ ಮಾಡೋಣ, ಅದರಿಗೋಸ್ಕರ ವಾಲ್ಯೂಮ್ ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡೋಣ. ಏರಿಯಾ ಇಂಟು ಥಿಕ್ನೆಸ್ ವಾಲ್ಯೂಮ್ ಆಗುತ್ತೆ. ಕೊಟ್ಟಿದ್ದ ಏರಿಯಾ ಥಿಕ್ನೆಸ್ 0.01 ಇಂಟು 10 to the power minus 3 ಮೀಟರ್ ಆಗುತ್ತೆ. ಮಿಲಿಮೀಟರ್ ಅಂದರು. 80 ಸೆಂಟಿಮೀಟರ್ ಅಂದರು 80 ಇಂಟು 10 power minus 2 ಸೆಂಟಿಮೀಟರ್ ಸ್ಕ್ವೇರ್ ಆಗುತ್ತೆ. ಇಫ್ ಐ ಕನ್ವರ್ಟ್ ದಟ್ ಇಂಟು ದಿಸ್, ಓಕೆ, ಇದನ್ನು ಕೂಡ ಸೆಂಟಿಮೀಟರ್ಗೆ ಮಾಡಿಬಿಡೋಣ, ಯಾಕಂದರೆ ಡೆನ್ಸಿಟಿ ಅನ್ನೋದು ಗ್ರಾಮ್ ಪರ್ ಸೆಂಟಿಮೀಟರ್ ಕ್ಯೂಬ್ ಇದೆ. ಸೊ ಏರಿಯಾ 80 ಇಂಟು 10 power minus 2 ಸೆಂಟಿಮೀಟರ್ ಸ್ಕ್ವೇರ್ ಇರಲಿ. 0.01 ಮಿಲಿಮೀಟರ್ ಅಂದರೆ 0.01 ಇಂಟು 10 power minus 3 ಇಂಟು ಮತ್ತೆ ಸೆಂಟಿಮೀಟರ್ ಕನ್ವರ್ಟ್ ಮಾಡಿದ್ರೆ 10 power 2 ಆಗುತ್ತೆ. therefore 10 power 2 ಇದು ಕ್ಯಾನ್ಸಲ್ ಆಗಿ 10 power minus 3 ಇಂಟು 80 ಇಂಟು 10 power minus 2 ಅಂದರೆ 80 ಇಂಟು 10 power minus 5. Density is weight by volume. 10.5 equals weight value ಗೊತ್ತಿಲ್ಲ. Volume is 80 into 10 to the power minus 5. So 10.5 into 80 into 10 power minus 5 is weight. ಇಷ್ಟು weight ಬಂದಿದೆ. ಇಷ್ಟು weight ಬಂದಿದ ಮೇಲೆ ನಾವೇನು ಮಾಡಬೇಕು? weight is 10.5 into 80 into 10 power minus 5. 108 ಅನ್ನೋದು ಅದರ ಇದಾಗತ್ತೆ ಅಂದರೆ ಈಕ್ವಿವೆಲೆಂಟ್ ಮಾಸ್ ಆಗತ್ತೆ 96,500 ಇಂಟು ಕರೆಂಟ್ ಪಾಸ್ ಮಾಡಿದ್ದು ಗೊತ್ತಾಗ್ತಾ ಇಲ್ಲ, ದೇರ್ ಫೋರ್ ನಾವು ಅದನ್ನು ಕೊಟ್ಟಿದ್ದಾರೆ 1.56, ಟೈಮ್ ಗೊತ್ತಿಲ್ಲ, ಸೊ ವಿ ನೀಡ್ ಟು ಕ್ಯಾಲ್ಕುಲೇಟ್ ದ್ಯಾಟ್. ಸೊ ಉಳಿದಿದ್ದೆಲ್ಲ 10.5 ಇಂಟು 80 ಇಂಟು 10 to the power -5 ಇಂಟು 96,500 ಡಿವೈಡೆಡ್ ಬೈ 108 ಇಂಟು 1.56 ಈಕ್ವಲ್ಸ್ ಟಿ ಟೈಮ್ ಅನ್ನೋದು ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡಬೇಕಾಗತ್ತೆ. ಇದೆಲ್ಲ ಯೂಸ್ ಮಾಡಿ ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡಿದರೆ ನಮಗೆ ಸುಮಾರು ಇದರಲ್ಲಿ 10 ಟೈಮ್ಸ್ ಅಂತ ತೆಗೆದು ಬಿಟ್ಟರೆ 8 ಟೈಮ್ಸ್ ಇಲ್ಲಾಗತ್ತೆ. ಆ್ಯಂಡ್ ಉಳಿದಿದ್ದ ಕ್ಯಾಲ್ಕುಲೇಷನ್ ಮಾಡಬೇಕು 96,500 ಇದರಲ್ಲಿ 10 to the power -3 ಬರುತ್ತೆ, 965,156. ಇದರಲ್ಲಿ 6.1 times. If I write 10 power -2 here, that is 10 power -1, 48. Option number 4, 48, 481 ಬರುತ್ತೆ. 80th question. The slope in the log k versus 1/t curve is, ಅದು 5.42 ಆಗಬಾರ್ದು, -5.42 ಇರಬೇಕು ಅದಕ್ಕೆ. ಆವಾಗ -EA ಬೈ ಲಾಗ್ ಅಲ್ವಾ 2.303 r ಸ್ಲೋಪ್ ಇದು, ದಟ್ ಈಸ್ ಈಕ್ವಲ್ ಟು -5.42 ಇಂಟು 10 ಪವರ್ 3. ಆಕ್ಟಿವೇಶನ್ ಎನರ್ಜಿ ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡಬೇಕು. ಮೈನಸ್ ಮೈನಸ್ ಕ್ಯಾನ್ಸಲ್ ಆಗುತ್ತೆ. 5.42 ಇಂಟು 10 ಪವರ್ 3 ಇಂಟು 2.303 ಇಂಟು 8.314, ಇದು 19.147 ಇಂಟು 5.42 ಮಾಡಬೇಕು. 19.15 ಅನ್ಕೋ, 5.42 ಇಂದ ಮಲ್ಟಿಪ್ಲೈ ಮಾಡಿದರೆ 3830 ಬರುತ್ತೆ. 7660 ಬರುತ್ತೆ. 5 5s are 25, 5 1s are 5, 7, 95, 3, 14, 19, 17. This is what we get. So it is 104, 103.7 ಅಂದರೆ 104 ಕಿಲೋಜೂಲ್ ಬರುತ್ತೆ. ಯಾಕಂದರೆ ನಂಬರ್ ಯು ಕ್ಯಾನ್ ಸಿ 4 digits ಅಷ್ಟೇ ಇಲ್ಲಿ ಹಾಕಿರ್ತೀನಿ, therefore you will get 104. ನೆಕ್ಸ್ಟ್ ಕ್ವೆಶ್ಚನ್ ಏನಂದರೆ ವಿಲ್ ಗೆಟ್ 81, ಯಾಕಂದರೆ ಇಂಟು 10 power 3 ಒಂದಿದೆ, ಕಿಲೋಜೂಲ್ ಬರುತ್ತೆ ಆನ್ಸರ್. 81st question. For a reaction A to B, it was found that the rate of a reaction doubles when the concentration of A is increased by 8 times. The order of reaction is ಅನ್ನೋದು ಕ್ವೆಶ್ಚನ್. ಇದು ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡಬೇಕಂದ್ರೆ ರೇಟ್ ಆಫ್ ದ ರಿಯಾಕ್ಷನ್ ಕೆ ಇಂಟು ಕಾನ್ಸಂಟ್ರೇಷನ್ ಆಫ್ ಎ ಇಂಟು ಪವರ್ ಎನ್ ಅನ್ಕೊಂಡ್ರೆ, ರೇಟ್ ಆಫ್ ದ ರಿಯಾಕ್ಷನ್ increases by 2 times when this increases by 8 times. Therefore 2r equals 8 power n into k into a power n. 2r equals 8 power n into r. 2 equals 8 power n. 2 equals 2 cube power n, 2 power 3n. Therefore 3n, 1 equals 3n, n equals 1 by 3. Option number 4 is correct answer. 82. Which reagent cannot be used to prepare an alkyl chloride from alcohol? Barely NaCl ತಗೋಬಾರ್ದು, ಅದರ ಜೊತೆಗೆ ಸಲ್ಫ್ಯೂರಿಕ್ ಆಸಿಡ್ ತಗೋಬೇಕು. therefore option 2 is not possible. ಲೂಕಸ್ ಟೆಸ್ಟ್ ಅನ್ನ ರೂಮ್ ಟೆಂಪರೇಚರ್ ಹತ್ರ ಪ್ರೈಮರಿ ಆಲ್ಕೋಹಾಲ್ ಕೊಡಲ್ಲ, ಸೊ ಫಸ್ಟ್ ಆಪ್ಷನ್. ಎಸಿಟಿಕ್ ಆಸಿಡ್ಗೆ ಕ್ಯಾಲ್ಸಿಯಂ ಆಕ್ಸೈಡ್ ಮತ್ತು ಸೋಡಿಯಂ ಹೈಡ್ರಾಕ್ಸೈಡ್ ಹಾಕಿದರೆ ಅದು ಕಾರ್ಬಾಕ್ಸಿಲೇಷನ್ ಆಗಿ ನಮಗೆ ಮೀಥೇನ್ ಬರುತ್ತೆ. ಆ ಮೀಥೇನ್ಗೆ ಮತ್ತೆ ಬ್ರೋಮಿನ್ ಇನ್ ಸನ್ಲೈಟ್ ಹಾಕಿದರೆ ಮಿಥೈಲ್ ಬ್ರೋಮೈಡ್ ಬರುತ್ತೆ. ಮಿಥೈಲ್ ಬ್ರೋಮೈಡ್ಗೆ ಸಿಲ್ವರ್ ಸೈನೈಡ್ ಹಾಕಿದರೆ ಮಿಥೈಲ್ ಐಸೋಸೈನೈಡ್ ಬರುತ್ತೆ. therefore option number 3 is correct answer. ಡೈಮಿಥೈಲ್ ಫಾರ್ಮಮೈಡ್ ಅನ್ನುವ ರಿಯೇಜೆಂಟ್ ಅನ್ನು ಯೂಸ್ ಮಾಡಿದರೆ ಸಾಲ್ವೆಂಟ್ ಅನ್ನ, ಡೈಮಿಥೈಲ್ ಫಾರ್ಮಮೈಡ್ ಇಸ್ ಅ ಪೋಲಾರ್ ಅಪ್ರೋಟಿಕ್ ಸಾಲ್ವೆಂಟ್, ಅಂದ್ರೆ SN2 ಮೆಕ್ಯಾನಿಸಮ್ ಅನ್ನ ಇಂಡಿಕೇಟ್ ಮಾಡ್ತಾ ಇದೆ. ಅಂದ್ರೆ ಇನ್ವರ್ಷನ್ ಆಗಿರಬೇಕು. ಇನ್ವರ್ಷನ್ ಆಗಿರ್ಬೇಕಂದ್ರೆ Cl ಪ್ಲೇಸ್ ನಲ್ಲಿ NC ಬಂದು ಅದು ರಿವರ್ಸ್ ಆದರೆ ಸೆಕೆಂಡ್ ಆಪ್ಷನ್ ಬರುತ್ತೆ. ಆಪ್ಷನ್ 2 ಇಸ್ ಕರೆಕ್ಟ್ ಆನ್ಸರ್. 86. Which of the following statements is not correct? Nucleoside is an N-glycoside of heterocyclic base. Correct ಅದು. Heterocyclic base ಮತ್ತು ಗ್ಲೂಕೋಸ್ ರೈಬೋಸ್ ಕನೆಕ್ಟ್ ಆದರೆ we call it a glycoside. It is correct. Nucleotides are phosphoesters of nucleosides. ಇದು ಕೂಡ ಕರೆಕ್ಟೇ. The structure of ribose sugar is ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ, ಬಟ್ ಅಲ್ಲಿ ಕೊಟ್ಟಿದ್ದು ಡಿಯಾಕ್ಸಿ ರೈಬೋಸ್, ಸೆಕೆಂಡ್ ಪೊಸಿಷನ್ ಅಲ್ಲಿ OH ಇಲ್ಲ, ಸೊ ಅದು ತಪ್ಪಾಗುತ್ತೆ. 87th question. CH3 para, I mean CH3, CH2, CH2, NH, CH3 ಅದು ಕೊಟ್ಟಿದ್ದಾರೆ. They look like they're metamers because length of alkyl chain is different between nitrogens. Therefore we need to say they are metamers. 3 is correct. Alcohol and ether ಕೊಟ್ಟಿದ್ದಾರೆ. That is actually functional isomers. And then alcohol and secondary alcohol ಕೊಟ್ಟಿದ್ದಾರೆ. I mean they are position isomers and the rest are chain isomers. Therefore option 1 is correct. An inorganic compound X on treatment with concentrated sulfuric acid produces brown fumes. Andre it should contain a nitrate ion and it produces dark brown ring, so definitely nitrate ion. And also compound X gives precipitate Y when its solution in dilute HCl is treated with H2S. Andre second group nalli barta ide. Then the precipitate Y on treatment with concentrated nitric acid followed by excess of ammonium hydroxide further gives deep blue color, andre copper barutte, copper tetra amine. Option number 1 is correct answer. 50 ml 0.5 molar oxalic acid anna 25 ml sodium hydroxide ಗೆ ನ್ಯೂಟ್ರಲೈಸ್ ಮಾಡಲಿಕ್ಕೆ ಯೂಸ್ ಮಾಡಿದರೆ amount of sodium hydroxide in 50 milliliter of the given sodium hydroxide solution ಎಷ್ಟು? ಫಸ್ಟ್ ನಾವು ಇದಕ್ಕೂ ಕೂಡ A1 M1 V1 ಈಕ್ವಲ್ಸ್ A2 M2 V2 ಅನ್ನುವ ಫಾರ್ಮುಲಾನ ಹಾಕೋಣ. ಹಾಕಿದರೆ ನಮಗೆ A1 ಅಂದರೆ ಆಕ್ಸಲಿಕ್ ಆ್ಯಸಿಡ್ನಲ್ಲಿ 2 ಬರುತ್ತೆ, M1 ಅಂದರೆ 0.5 ಬರುತ್ತೆ, ವಾಲ್ಯೂಮ್ 50 ಬರುತ್ತೆ. A2 ಅಂದರೆ ನಮಗೆ ಸೋಡಿಯಂ ಹೈಡ್ರಾಕ್ಸೈಡ್ಗೆ 1 ಬರುತ್ತೆ, ಮೊಲಾರಿಟಿ ಗೊತ್ತಿಲ್ಲ, ವಾಲ್ಯೂಮ್ 25 ಅಂದರೆ ಇದು ಆ್ಯಕ್ಚುಲಿ 25 ಆಗುತ್ತೆ. 25 ಕ್ಯಾನ್ಸಲ್ ಆದರೆ ಮೊಲಾರಿಟಿ 0.2 ಆಗುತ್ತೆ. ಮೊಲಾರಿಟಿ ಈಕ್ವಲ್ಸ್ ಮಾಸ್ ಬೈ ಮೋಲಾರ್ ಮಾಸ್ ಇಂಟು 1000 ಬೈ ವಾಲ್ಯೂಮ್ ಅನ್ನೋದು ಫಾರ್ಮುಲಾ. ಮೊಲಾರಿಟಿ ಇಸ್ 0.2. ಮಾಸ್ ಆಫ್ ದ ಸಬ್ಸ್ಟೆನ್ಸ್ ಅನ್ನೋದು ಗೊತ್ತಿಲ್ಲ, ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡಬೇಕು. ಸೋಡಿಯಂ ಹೈಡ್ರಾಕ್ಸೈಡಿನ ಮೋಲಾರ್ ಮಾಸ್ 40 ಆಗುತ್ತೆ. 1000 ಡಿವೈಡೆಡ್ ಬೈ ವಾಲ್ಯೂಮ್, ವಾಲ್ಯೂಮ್ ಇಸ್ 50 milliliter and hence what we need to consider is ಇದು 200, 4 5s are 22,000 ಆಗುತ್ತೆ. 2 times 2 is this 0.1. 4 ಆಗತ್ತೆ, 4 ಗ್ರಾಮ್. ಯಾಕಂದರೆ ಇದು 2 ಈ ಕಡೆ ಬಂದರೆ ಇಟ್ ಬಿಕಮ್ಸ್ 4 ಗ್ರಾಮ್ ಬರುತ್ತೆ. ಆಪ್ಷನ್ 1. Assertion. A bright silver mirror is produced during the warming of an acetylene with freshly prepared ammonical silver nitrate solution. When we consider bright silver mirror ಬರಲ್ಲ, silver acetylide ಅನ್ನುವ ವೈಟ್ ಪ್ರೆಸಿಪಿಟೇಟ್ ಬರುತ್ತೆ ಟೊಲೆನ್ಸ್ ರಿಯೇಜೆಂಟ್ ಒಂದಿಗೆ. HC ಟ್ರಿಪಲ್ ಬಾಂಡ್ CH ನಲ್ಲಿ ಇದು ಎಸಿಡಿಕ್ ಅಲ್ವಾ, ಸೊ ಸಿಲ್ವರ್ ನೈಟ್ರೇಟ್ ಅನ್ನ ಯೂಸ್ ಮಾಡಿದರೆ HNO3 ಹೋಗ್ಬಿಟ್ಟು ಸಿಲ್ವರ್ ಎಸೆಟಲೈಡ್ ಅನ್ನುವ ವೈಟ್ PPT ಬರುತ್ತೆ. ಸಿಲ್ವರ್ ಮಿರರ್ ಆಲ್ಡಿಹೈಡ್ಸ್ ಬರುತ್ತೆ, ದೇರ್ಫೋರ್ ಇದಕ್ಕೆ ಬರಲ್ಲ. ರೀಸನ್, a bright silver mirror is produced due to the reduction of Ag plus 2 to Ag. ಅದು ಕರೆಕ್ಟೇ, ಬಟ್ ನಾಟ್ ದ ಫಸ್ಟ್ ಸ್ಟೇಟ್ಮೆಂಟ್. ಸೊ ಅಸರ್ಷನ್ ಇಸ್ ಫಾಲ್ಸ್, ರೀಸನ್ ಇಸ್ ಟ್ರೂ. ಇಲ್ಲಿಗೆ ಇದು ಮುಗೀತು. ಉಳಿದಿದ್ದು 10th ಗ್ರ್ಯಾಂಡ್ ಟೆಸ್ಟ್ಗೆ ಮತ್ತೆ ಭೇಟಿಯಾಗೋಣ. ನಾಳೆ 9th ಗ್ರ್ಯಾಂಡ್ ಟೆಸ್ಟ್ ರಾಮರಾಜ್ ಸರ್ ವಿಲ್ ಡೂ ಇಟ್.
Okay, good evening all of you. Now today paper discussion. So one by one amma. Now first one. The speed of the boat is 5 km per hour in still water. The width of the river is 1 km along the shortest possible path. Shortest possible path means a boat is moving like this one. A boat is moving like this one. The water is flowing like this. What is the net velocity, amma? This is the hypotenuse, so this is the one side. What is the remaining value? VBR square minus VR square. Shortest possible path means boat is moving like this one by the water flow boat is moving along perpendicularly. That's why shortest path means like this one. What is the time distance? D is nothing but width of the river. Velocity VBR square minus VR square. This is the formula. What is the time value given? 15 minutes. 15 minutes, take them in hours. 15 by 60 hour. What is the distance, 1 kilometer by root of VBR 5 square 25 minus VR square, where VR is nothing but velocity of the river. This value 4 times, so cross multiplying them and squaring. Therefore, VR square is equal to 9. Therefore, VR is equal to 3 km per hour. 3 km per hour. That is the first question. Second question. The initial velocity U bar is equal to I plus 2j. That's why, what is the equation? Already we know that this value is similar to that of U cos theta I cap U sin theta j cap gamma. So that's why U cos theta value 1 no, U sin theta value 2. U cos theta value 1 no, U sin theta value 2. That's why tan theta value is equal to how much sin theta by cos theta, like 2. Okay, but what is the equation? Already we know that y is equal to ax minus bx square. What is the equation of the parabola? Y is equal to ax minus bx square, where a is equal to tan theta, where b is equal to g by 2u square cos square theta. Already we know that formula. What is the value of tan theta? Tan theta value 2, g value 10 by 2. What is the u cos theta? U cos theta like 1 ega. U cos theta whole square kada like 1. How many times 5 times. Therefore, y is equal to a 2x minus b value 5 x square. Y is equal to 2x minus 5x square, like first option. Now next one, third problem. In the graph, like time and the position graph, like this one. So this value here 4, this value is 2, this value is 4 Here remaining value is no need. Body mass value 0.1 kg. 0.1 kg. So that's why what is the impulse at 2 seconds? Impulse is also known as a change in momentum. Change in momentum means m into v minus u. M into v minus u. Mass value 0.1. Right here 0 to 2 seconds is velocity value same. And like initial velocity. How to find the initial velocity? Slope. What is the tan theta? Tan theta opposite side by adjacent side. Like 4 by 2, like 2. Initial velocity 2. What is the final velocity? We can check them in this 2 seconds means like this one. 2 to entire the path like this one gara. Position is not changing 2 seconds position, 4 seconds position same and the particle at rest. Because of position is not changing. Particle at rest means final velocity 0. So that's why our answer minus 0.2. Minus 0.2 amma. As the initial gamma up to initial to up to below 2 seconds particle position is changes. They having the velocity. 2 to the 2 seconds to the entire the motion the particle at rest like final velocity 0. So that's why body initial velocity amma, body final velocity 0 namma. That's where answer we get minus 0.2. Now next one amma, question number 5 First we can explain the some difficulty models amma. That's where listen carefully. This is the one particle velocities are like this. Velocity of the particle P amma has the velocity is 10 angle 30 degrees. Velocity of the Q is nothing but 6 here angle 30 degrees. What is the separation between them amma? 20 centimeter. Sorry amma, 20 meters. That's why what is the calculate the angular velocity of P with respect to Q. Angular velocity of P with respect to Q. Now this V value we can resolve them like this one and this one. 10 cos theta, 10 sin theta. Sin theta means 1 by 2, 5. Again this 6 value resolve the two component like this one another component. 6 cos theta, 6 sin theta like 3. So as finally we can observe them, this one amo 3, this one amo 5. In between the gap amo 20. What is the angular velocity amma? V is equal to R omega. Omega is nothing but V by R. Change in velocity by distance. What is the change in velocity? Both are in opposite direction amma. 3 minus of minus 5. Like 8. Change in velocity by like distance amma. Change in velocity by like perpendicular distance. 4 2s are 4 5s are 2 by 5 like 0.4 radian per second. 0.4 radian per second. So the angular velocity omega is equal to change in velocity by distance between them. Change in velocity like this one like this one amma. Perpendicular, both are perpendicular. Change in velocity is nothing but opposite. Take them minus of minus plus amma and distance amma 20. We get them. Problem number 7 number. This is the 2 meters amma. This is the 3 meters. Here amma one axis is there amma. This axis amma like a center of mass amma is the given center of mass ra. Overall distance amo A. That's why what is the moment of inertia about an axis passing through the center of mass. Okay now listen. 2 masses amma 2m, this value amma 4m ra. Like this is the origin amma. How much distance amma A ga? What is the origin amma? Sorry, what is the coordinate amma? A comma 0. How to find the center of mass amma? Xm is equal to m1 x1 2m into 0 0 3m into A by m1 plus m2. Already we know that m1 x1 plus m2 x2 by m1 plus m2. m1 x1 0, m2 x2 like this one by m1 plus m2. M value cancel amma, we get 3a by 5. Like what is the center of mass location amma? 3a by 5. X value 3a by 5. Remaining value amma 2a by 5. 2a by 5. Therefore, the moment of inertia is equal to mr square. M value amma 2m. R square 3a by 5 whole square. This mass value amma 3m. Distance amma 2a by 5 whole square. Moment of inertia mr square. That's why both are adding we get 6 by 5 mr square. 6 by 5 ma square amma. This is the center of mass finding. Already previous one model is there amma. Same model. 6 by 5 ma square. Next one, Amma. Problem number 9. Here, Amma, PV graph is given. Volume and pressure. So, D, E, F. Here, Amma, 3. This value, Amma, 7. 200. 600. The total work done by the gas D to E and E to F. D to E and E to F. What is the work done? And a simple like this one area. So that the total area is nothing but half into base into height half into base value 7 minus 3 4. Height value 600 minus 200 400. That's why 800 joules. Problem number 10. In the figure, the intensity of the waves arriving at D from the two coherent sources S1 and S2 is I0. The wavelength of the wave is lambda. What is the resulting intensity at the point D? Already we know that the resulting intensity is nothing but 4 I0 cos square 5 by 2. Because of both having the same intensity, I0 is given. That's where what is the resulting intensity? 4 I0 cos square 5 by 2. 4 I0 cos square 1 by 2 into 5. 5 is nothing but phase difference. What is the relation between the phase difference and the path difference? 2 pi by lambda into delta x. Phase difference is equal to 2 pi by lambda into delta x. These two are cancelled. 4 I0 cos square 5 by lambda. Lambda value is given how much 4. Lambda value 4. What is the path difference What is that path difference? Now listen. Path difference like these two are the sources. This is the point P. This is the 3 meters. So this is the 4 meters. What is the meaning of path difference? S2P minus S1P. 3, 4 automatically this one 5 vega. What is the S2P 5. What is the S1P 4. What is this value 1. Like 1. 4 I0 cos 45 1 by root 2 whole square 1 by 2. We get 2 I0. What is the path difference? Already we know that these waves are reaching here, these waves are reaching here. What is the path difference? S2P minus S1P. 3, 4 gamma automatic 5. S2P and 5, S1P and 4. We get 1. The delta x value 1 substituting here, we get 2 I0. Now problem number 11. This is the one Gaussian surface Gaussian surface Here one charged wire This is the charged wire whose linear charge density lambda. This is the charged wire. This is the center. Here R by 2. One line here Just what is asking flux? The total flux is equal to Qn by epsilon naught. What is the meaning of charge? Lambda into L. Linear charge density lambda is there epsilon naught. Just we can find this length problem is over. That length value substituting here we get the answer. lambda by epsilon naught of, how to find that length? Already we know that this value r by 2 is given, amma. This one name radius. What is this value, amma? This value r by 2 gara. What is the value of this one? Take them theta. Cos theta. Cos theta is nothing but how much, amma? Adjacent side by hypotenuse. And y 1 by 2. Theta value 60. Theta value 60. Automatically this value 30. And this value 30 ga. Resolve them. R value gamma. R value is nothing but this one ga. And here what is this value? R cos 30. This value R gamma. Resolve them. R cos 30. What is the total length, amma? That the total length L is equal to R cos 30, R cos 30, 2R cos 30, root 3 by 2, like root 3R. Root 3R. Cos theta value adjacent side by hypotenuse 1 by 2. Theta value 60, automatic 30 because of here 90 ga. R value 30 degrees R cos 30. R value horizontal 30 gara R cos 30. What is this total length 2R cos 30. We get root 3R. That length value substituting here, we get the flux. Problem number 12. The charge on the condenser of capacitance 2 microfarad capacitor. This one capacitor, here 2 capacitors. So that's why what is the charge on the 2 microfarad capacitor? Now listen, amma. Here 8 volts, amma. Here also 8 voltage. And here also 8 volt, amma. Here also 8 volts, right? So up to here 8 volts. For example, these two are in parallel gara. What is the value of 5 microfarad? 5 microfarad. Right, amma. So these two are connected in series, amma. In series lo charge value same ra. In series lo charge value same and the voltage is different gamma. How to find that voltage ra? In this one. Q is equal to CV, amma. In this one, amma, charge value same, amma, because of 3 and 5 are in a series, gamma, same. And the voltage inversely proportional to capacitance. Like V1 by V2 is equal to C2 by C1. What is the value of C2, amma? 5. C1 value 3. Therefore, V2 value is equal to how much, amma? V2 value is how much here? 3 by total parts 8. 5 plus 3, 8. What is the net voltage is given, amma? 8. That's why how much 3 volts. And the V2 value 3 volts means, amma, here 3 volts. Total 8 lo 3 over gara, here 5 volts. Already we know that these two are in parallel gamma, voltage same. Here are 3 volts, here are 3 volts. What is this charge, amma? Q is equal to CV. C value 2, voltage value 3. That's why 6 microcoulombs. 6 microcoulombs. Ma, these 3 microfarad, 5 microfarad, both are series. Series lo charge same voltage inversely proportional to capacitance, amma. V1 by V2 is equal to C2 by C1, amma. C2 value 5 C1 value 3. Total how many parts 8 parts. 8 parts, amma, both are adding how much voltage 8 and compulsory ga V1 value nemo 5 volts V2 value nemo 3 volts Both are in series gara, voltage value same therefore Q is equal to CV. That's why we get that 6 microcoulombs. What is the charge on the 3 microfarad? Again Q is equal to CV, 3 into 3, 9 microcoulomb. 9 microcoulomb in case of the 3 microfarad. Now next one, problem number 13. In a problem number 13, this is the time, this is the current I like microamperes. Here one graph is given like this, 2, 6, 8, this value 5, this value 15. What is asking charge flowing through it from 2 to 8. 2 to 8 how much of the charge is flowing through it. What is this graph IT graph charge, current is equal to charge by time, charge is equal to I into T. Current to time both are multiple gara, that's why I take the area. 2 to 6 kadamma like area. The area of the current on a time graph gives the charge Take them a rectangle take them, this is the trapezium. Both areas are we get the charge. Or this is the rectangle gara, this one one rectangle, one triangle. Otherwise also we get the same answer. That's where we can check them 40 microcoulombs. Now problem number 16 Problem number 16. Match the following here one side like current values are given that corresponding RMS values. Here current value x naught sin omega t. That's why RMS current is equal to I naught by root 2 gamma. What is the I naught place x naught by root 2. That's why A key second point is the correct one Mari second one x naught sin omega t cos omega t gamma. Here multiplying with 2 numerator and denominator. X naught by 2 sin theta cos theta sin 2 theta. Sin 2 theta. Again the same relation one What is the value of I naught maximum current like x naught by root 2 ne kada. Therefore IRMS is equal to I naught root 2. I naught by root 2. I naught plus x naught by 2 into root 2. So that one second one having the third option. Then automatically remaining one is nothing but first option gamma. Next one problem number 18. A photon is accelerated through a voltage V has the de Broglie wavelength is lambda. For a alpha particle accelerated through the same voltage what is the de Broglie wavelength? Lambda is equal to h by 2m charge into voltage. Here voltage value same 2 value constant and h value is also constant Lambda is inversely proportional to mass into charge First one proton, second one alpha particle. Proton and alpha particle. Inversely mass of the proton, charge of the proton. Alpha particle, charge of the alpha particle. Already we know that mass of the proton m, charge of the proton E. Alpha particle 4m. charge ఏమో 2e. For proton ఏమో lambda, we don't know about the alpha particles. mu, e, mu cancel. 4 value take them outside ra 2. This value name √2. Therefore, lambda of the alpha particles ra lambda by 2 √2. Next one, amma. Problem number 20. So in the given resistance bridge, amma, as shown, amma, there is no current flowing. There is no deflections in the galvanometer across B and D. In B and D, amma, there is no galvanometer deflection, means the circuit is in balanced. That's why what is the value of R? Already we know that when the bridge is balanced, P by Q is equal to R by S. P value is 100. Q value is 200. Next, R value, R and 100 both are in parallel. P1, P2 by P1 plus P2. Like R1, R2 by R1 plus R2. S value name 40. So this value 2 times ra. This value ra again 20 times ra. Cross multiplying. 20 is equal to 100 R by 100 plus R. This 20 ra 5 times. Cross multiplying, 100 plus R is equal to 5R. 4R is equal to 100. Therefore, R is equal to 25 ohms. Bridge is a balanced, P by Q is equal to R by S. (23) A glass thickness 3 centimeter is placed on the ink mark of the piece of paper. This is the paper, this is the ink mark. One glass slab is there, how much centimeter? 3. For a person is looking at the mark at a distance 5 centimeter above it. This is the person 5 centimeter, already 3 over gara. This value like 2 and total 5. The distance of the mark will appear to be 4 centimeter, and total this person see this one mark like mark how much distance 4 centimeter. 8 2 as it is actual 3 now another take them this is the object, this is the image. 4 centimeter this one 2 and overall 4 object 5 centimeter. That's why what is the refractive index? Already we know that apparent depth is equal to real depth by mu. What is the apparent depth? 2. Real depth overall 3 by mu. Therefore, mu is equal to 3 by 2. Mu is equal to 3 by 2. Now, problem number 25. When this is the circular ring, charged ring, that is the plus Q. Radius is R. At what distance, X value like √3R. What is the field, what is the electric field at a distance? That electric field at a distance is equal to K 1 by 4 pi epsilon naught Qx by R square plus X square whole power 3 by 2. The electric field at a distance x K 1 by 4 pi epsilon naught Qx by R square plus X square whole power 3 by 2. We can check them ra. X value √3 √3 simplify them ra, we get third option. #27. A proton is projected with a speed of 2 into 10 power 6 meter per second at an angle 60 degrees with the x-axis. Magnetic field is applied along the y-axis. That's why what is the path of the proton? What is the path of the proton? Like this one y-axis that is in this direction the magnetic field is there. What is the value of magnetic field 0.104 Tesla. One proton is projected with a speed 60 degrees to the x-axis. This is the x-axis like particle is projected with a speed u 2 into 10 power 6. How much angle 60 degrees with the x-axis. That's why what is the path of the proton? Now listen this value in this side u cos theta. This side u sin theta. This value magnetic field perpendicular, like velocity perpendicular. This value magnetic field parallel, like velocity like parallel. Right? How to find that path? This component is trace the magnetic field in circle. This component is moving like as it is. That's why what is the path? Like a helical path. So in helical path, what is the radius? R is equal to general formula mv sin theta. V is nothing but perpendicular velocity like this one. What is that perpendicular velocity? mu cos 60 by. So R is equal to mv by bq. Sorry, R is equal to mv by bq. What is the perpendicular velocity? U cos 60 by bq. We get the radius. How to find the time period? Time period is nothing but distance between them. R is equal to 2 pi m by bq. Time period is equal to 2 pi m by bq. The time period is independent of the velocity. That's where time period is equal to 2 pi m by bq. And R is nothing but mv by R is equal to mv by bq. V and the perpendicular velocity. U cos 60 by bq. That's where we get the radius and the time period. Path is helix. Helical path. Already we know that these formulas. #29. A circuit consisting of the inductance and the resistance are joined in a series by the 220 volts. Inductance and a resistor are in a series by 250 volts. It draws a current 10 amperes. It is used in the circuit has a power value. Power value 1500 watts. That's why what is the wattless current? Wattless current. What is the meaning of wattless current? The wattless current is nothing but I sin phi. Wattless current is nothing but I sin phi, where phi is the phase difference between the voltage and the current. How to find that one? First, power is equal to VI cos phi. Power value 1500, voltage value 250, current value 10 cos phi. 2 zeros cancel amma. Cos phi is equal to 15 by 25 amma, like 3 by 5. Then cos phi value 3 by 5 gara, then automatic sin phi value 4 by 5. Therefore, wattless current, I sin phi gamma. What is the current value? 10. Sin by under 4 by 5 amma. We get 8 amperes. 8 amperes. Wattless current means amma, I sin phi. $30. The linear density mu is equal to 1.3 into 10 power minus 4 kg per meter. Therefore, the transverse wave is propagating in a string like 0.021 sin x plus 30t. That's why what is the tension in the string amma? Already we know that. What is the velocity of the wave amma? Omega by k ga. Omega value 30 amma, k value 1 amma. Therefore, velocity is nothing but 30 meter per second. The velocity of the transverse wave in a rope is equal to V is equal to root of T by mu. Squaring on both sides amma, therefore tension is equal to V square mu. V square value 900 mu value 1.3 into 10 power 14 we get the tension. We can check them we get the tension. $31 amma. A tuning fork producing a beats per second with 2 argon pipes having the length 30 centimeter and 31 centimeter amma. How many beats are produced? 4 beats are produced amma. That's why what is the frequency of the tuning fork? The number of the beats n1 minus n2 how many beats are there 4 beats That's why that 2 argon pipes lengths 30 centimeter and 31 centimeter. 2 argon pipes lengths values are given So 2 argon pipes like this one. This is all the water is there Tuning fork So what is the frequency of the tuning fork like this one? So the frequency of the tuning forks whose length values are given 30 and 31 already we know that the velocity of the sound is equal to 2n into L2 minus L1 2n into L2 minus L1 Already we know that formula, velocity of the sound, V is equal to 2n into L2 minus L1. L1, L2 values are given So, argon pipe length value L1 and 2 given but we don't know about the velocity n value you can find this L value. How to this find But how to find this velocity of the sound like this one. What is the frequency of the first one V by 4 L1 minus V by 4 L2 is equal to 4. So take them V value common V value common like V by 4 value common L1 L2 here L2 minus L1 is equal to 4. So what is the value of V This V value take them here 16. This value simplifying we get the V value. That V value substituting in this equation we get the velocity of the sound. Velocity of the sound that V value substituting here we get the frequency. But lengthy derivation you can check them carefully. Just to give the idea. Already we know that velocity of the sound 2n into L2 minus L1. So where n is nothing but frequency of the tuning fork, V by 2 into L2 minus L1. We don't know about the velocity. But n1 minus n2 is equal to 4. What is the fundamental frequency in a closed pipe? V by 4L1. Second case V by 4L2 is equal to 4. Here V is the velocity of the sound gamma. That value simplifying by this one and substituting here we get the frequency. But time taking just I am giving the idea, we can try it . Now next one . 32. When a mass m is attached to the spring is extended by 0.2 meters . The mass m is a slightly additional extension and released. What is the time period? What is the time period? Already we know that initial some load is attached . M mass is attached . By this mass attached is pull them and release it . Mg value is equal to Kx . Mg is equal to Kx. That's why is asking the time period. What is the time period ? T is equal to 2 pi into root of m by k . Mg, G value 10, K, X value is how much is given? 0.2. That's why G value I can write as it is. What is the value of m by k? M by k 0.2 by G nega. 0.2 by G. But in bracket G value pi square is given 2 pi into root of m by k value 0.2 by G value as it is. Okay, 0.2 means 2 by 10, like 1 by 5. 1 by 5. G value pi square. Pi square take them outside. This pi value cancel. What is the remaining value? 2 by root 5. 2 by root 5 seconds. G value pi square. 0.2 means 1 by 5. Pi square take them outside, cancel. Remaining value 2 by root 5 seconds. Next one 34. 34. VRMS oxygen molecule is equal to that for hydrogen molecule . So that's why at 200 Kelvin hydrogen gas at 200. What is the oxygen temperature? VRMS is equal to root of 3RT by M. 3R value constant temperature by molecular weight. V1 by V2 is equal to root of T1 by T2, M2 by M1 over. But these two are same These two are same like 1A. This value 1 squaring on both sides Squaring on both sides. Now sorry values are same That one more step we can take them. VRMS is also same 3R value is a constant That's why temperature is directly proportional to molecular weight. Temperature is directly proportional to molecular weight. Therefore, T1 by T2 is equal to M1 by M2. Problem over. Because of RMS value is also same 3R value constant squaring on both sides T directly proportional to M. T1 by T2 is equal to M1 by M2. For hydrogen molecular weight 2 Other for oxygen came 32. Hydrogen temperature value is given amma, we get the temperature of the oxygen. We get the answer. దిగిపోతాం సార్. Now next one amma, problem number 36. A pan is filled with hot food amma, is cooled from 94 to 86 in a time 2 minutes, and a surrounding temperature 20, and how long it will cool from 71 to 69, how much time is there? Already we know that from Newton's law of cooling, theta 1 minus theta 2 by T K into theta 1 plus theta 2 by 2 minus theta naught. Already we know that. First case ra theta 1 no theta 2 time values amma, and theta naught emo surrounding temperature ra, one equation. Second case lo theta 1 no theta 2 ra, time value we don't know ra, that values are substituting ra another equation. Both are divide them ra, we get that time amma by the Newton's law of cooling. Just note the formulas amma, we can try it amma. $38. One long rod amma, this is nothing but one rod amma. This end one end is fixed amma. Other end cross sectional area actually face outside amma, that can be turned on the front view for understanding purpose. This one is the radius kadamma, right? Actually this upper end of the wire is twisted amma. You can twist it because of one end is fixed gamma, other end is twisted means amma, this part is reaches to like this one amma. How much of the distance is moved by the upper layer ra? X. What is the distance between the two layers ra? L kada. This upper part is reaches to in this one kada. This angle is nothing but a shearing strain. What is the shearing strain amma? Distance moved by the upper layer by distance between the two layers is known as shearing strain. X by L. Already we know that this is the radius amma, this is the angle theta ra. Already we know X is equal to R theta ga, R theta by L. Radius value is how much is given amma? 1 centimeter by 100. Theta value amma 0.8 radians by length amma 2 meters. So that's where 2 here amma 0.4 by 100, 0.004. 0.004 amma. Like that one is also a radians. Angle is always in radians. Next one amma 39. So this is the one spring ra. Here mass emo 2m, here emo mass m ra. Upper one amma spring, lower one emo string. Here amma rope is cut. That's why what is the acceleration of the 2m and m. Now listen amma, before cut ra m to m both are weight acting on downward gara, and both weight and 3mg is acting downward gara, and spring force is acting upward gara. Like mg is equal to kx kada, kx is equal to mg means ra how much weight is acting ra? 3mg. Kx is upward gara. What is the meaning of kx ra? Like 3mg nega. And overall ga upward ra how much force is acting ra? 3mg. Body total weight mg is acting spring force kx. Kx is how much? 3 mg. That 3 mg is also acting upward because of kx is equal to 3 mg. After cut, what happens? Once this one is cutting, this m value is falling with gravity. M is falling with gravity, like G is downwards. But now listen carefully. This is the 2m gara. Already in spring force, how much force is acting on upward? 3 mg nega. Body weight 2 mg ga. Which force is more? Up one kada. That's why upper one is moving upwards, some acceleration A. What is the net force? 3 mg minus 2 mg. Like mg is equal to ma. Mass value is how much? 2 ma. mo cancel A value g by 2 upward. This value g downward. So the answer is g by 2 upward and gmo downward. First option. $40. Problem number 40, amma. Two bodies A and B of equal mass are separated by a distance d. Then the force is F. Therefore, that F is equal to G m1 m2 by d square, amma. All are nothing but same mass. Next, now 50% of the mass is transferred from A to B, amma. Separation between them d by 2 ra. And again in between the gap liquid is there, amma. What is the gravitational force? That force F is equal to G. Already we know that in between the two masses, any medium is there, that is the independent. Because of capital G is the universal gravitational constant. It is independent of the medium. So that's where a liquid is leave it. 50% of the mass like m by 2 is transferred. What is the remaining m by 2 ga? M by 2 is reaches to another one gara. That value 3m by 2 ga. Distance value d by 2 whole square means d square by 4 ga. Right. 2 2s are 4. These values are cancelled. 3 value take them separate. G m by d square and f ga. Like 3f. $41. The velocity of the body v is equal to 10 minus 5t. That's why what is the average velocity of the body during the time interval of 0 to 2 seconds? Already we know that v average is equal to integral v dt by dt 0 to 2 seconds. Already we know that this formula, already this one is the same one is also complete in alternative current. V value 10 minus 5t dt 0 to 2. Integral dt t, upper limit minus lower limit like 2. That value 1 by 2. Integral 10 dt like 10t minus 5 t dt t square by 2. T value 2 seconds. T value 2 and 20. T value 4 and how much 5 into 4 20 by 2 10. Like 10 by 2 5 meter per second. The average velocity 5 meter per second. $43. The velocity of the electron in a second orbit of the sodium atom is v. Similarly, what is the in fifth orbit? velocity is equal to 2.2 into 10 power 6 of z by n. So directly proportional to z by n. V is the velocity amma, atomic number emo 11 ra, orbit emo 2. Second one value we don't know ra, atomic number 11a, but fifth orbit ra. So therefore V by V1 is equal to 11 cancel ra 5 by 2. Therefore V1 amma 2V by 5. $45. A square loop ra side A carrying the current I. What is the magnetic field at the center of the loop? Current I amma. Same current is passing through in a loop gamma. What is the magnetic field at the center? Now listen ra. By this one ra current inside. 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 So the net magnetic field is equal to how many wires are there? 4 into each. 4 into each. Okay ra. This total side A gara half part ra A by 2. This is the square gara. Angle amo 45 and 45. Therefore 4 into what is the formula of the each one? Mu naught I by 4 pi r. R value A by 2. Into sin alpha, sin 45 plus sin 45. 4 value cancel ra. 2 mu naught I by pi a. Sin 45, 1 by root 2 plus 1 by root 2, 2 by root 2. This root 2 value here root 2 times ra. 2 root 2 mu naught I by pi a. 2 root 2 mu naught I by pi a, like first option. Okay, Namma. Just only some remaining questions are the very lengthy models, Amma. Just you can try it, Amma. But maximum difficulty models are is explained. Remaining 1 or 2 step models, Amma. Just you can try it. 1 or 2 questions are the very lengthy, Amma. That is the beyond level, Amma. That questions are is leave it, Amma. Right. So next one, we can continue. Thank you, Amma.